Well, good afternoon, folks. It's 1 o'clock by my watch, Eastern Standard Time. I hope you can all hear and see me. My name is Mukarrab Qureshi, and I wanted to welcome you to ARS Sweet Potato Symposium, entitled Goodness Grows in the Carolinas, a symposium focusing on advances in sweet potato research and processing through public and private partnership. I'm the research leader here at Raleigh location, and my partner, Dr. Phil Beetle, also joins us from Charleston, South Carolina location. Just some housekeeping announcements, folks. This meeting is being recorded, and we'll do our best to make the recorded version available to you all after the meeting. Please be mindful that your chat can be viewed by all participants. You can view current speakers with slides using side-by-side view option. Also, you can use chat box function to post any questions you may have or to share any comments. And you can also raise your hand if you want to make a comment, and I'll do my best to recognize you. Folks, why sweet potato? As you know, sweet potato is a major commodity here in the state of North Carolina. Very soon we'll be reaching $1 billion in revenues from fresh and processed sweet potato. So it's no brainer that USDA-ARS pays full attention to supporting the research agenda for the betterment of this commodity. We have brought really who's who of our colleagues to help us establish an agenda and a program. Will essentially, the main intent of this symposium is for us as USDA-ARS researchers to share all the good things which our team and our collaborators do in supporting this commodity. We'll have essentially two main themes. One is share, and the second theme, which is the most important theme in my opinion, is to listen to our stakeholders, what you have to share with us so that we remain aligned and relevant to our stakeholder needs. I'm so grateful to the key program contacts. We are really fortunate to partner in this effort with North Carolina Sweet Potato Commission. Michelle Granger, who's the Executive Director of Sweet Potato, North Carolina Sweet Potato Commission, is with us. And thank you, Michelle, for supporting and co-hosting this symposium with us. Dr. Craig Yancho is a great collaborator, William Neal Reynolds, Distinguished Professor at NC State University. And Sandra Parker, who has been my partner in crime. You've probably received a million emails from her. She's really the brain behind this whole effort. Dr. Phil Waddle is from our Charleston U.S. Vegetable Lab. And Dr. Suzanne Johanningsmeyer from here, Raleigh location. She's our brilliant research food chemist. Folks, just a little bit of who we are. We are, of course, U.S. Department of Agriculture, headed by our Honorable Secretary Vilsack, who just got appointed by President Obama and confirmed as the second term, or actually this would be his third term, Secretary of Agriculture. Then Dr. Shawana Jacobs-Young, who's our ARS administrator, but at the moment she's acting USDA Deputy or Undersecretary of REE, which is Research, Education, and Economics Mission Area. ARS, folks, is divided into several areas across the country and around the globe. We fall, the North Carolina and South Carolina research operations fall under Southeast area, which is headed by Mr. Archie Tucker and very ably supported by two of our associate area directors, Dr. Alan Harris and Dr. Prasanna Gowda. I would now like to invite Dr. Harris, if she has joined us, to please make a few comments about who we are and what we are all about. Dr. Harris?
Good afternoon, everyone. I'm trying to see if is my video on?
Yes, we can see you, Dr. Harris.
Okay, all right.
And hear you as well.
Okay, well, thank you, Dr. Qureshi, for your introduction. And it's my pleasure to join you this afternoon and represent the Southeast area leadership team on behalf of Mr. Archie Tucker, who is the area director, and Dr. Prasanna Gowda, who, along with me, the two associate area directors. The Southeast area, as you could see on the map that Dr. Qureshi showed, is a very large one. In fact, we are the largest area within the Agricultural Research Service. And we have 66 research units spread over 8 states and Puerto Rico. We conduct research just about every research subject matter area in ARS. I'm very glad to see that this symposium is a joint effort between USDA-ARS locations in Raleigh, North Carolina, and Charleston, South Carolina, in partnership with the North Carolina Sweet Potato Commission. It's exciting to see this effort take place. It's a very good example of a public-private partnership. As the intramural research agency in USDA, the ARS mission is to solve agricultural problems and to work closely with our stakeholders to meet their needs in order to move the science forward with purpose. This symposium is based, as Dr. Qureshi said, on an overarching theme of share and listen. And we look forward to you all's input and feedback in advising us on how to serve you even better. Now, just a few facts. Besides the fact that I cook a mean, savory, roasted sweet potato dish, some highlights which sort of mark the reason for calling this symposium Goodness Grows in the Carolinas is the fact that North Carolina alone produced 55% of sweet potatoes grown in the United States in 2020. Very soon, the fresh and processed sweet potato market will be touching $1 billion, as Dr. Qureshi pointed out earlier, per year. North Carolina is #1, and both the state and South Carolina are among the top 10 sweet potato producing states in the country. 98% of the US sweet potato exports come from North Carolina. And the last but not least point is the US is the #1 exporter in the world, making North Carolina the #1 sweet potato exporter with 41% of the global sweet potato export market. So a very good reason to call this Goodness Grows in the Carolinas. This is why it is extremely important for USDA-ARS to support the sweet potato industry through a strong and sustainable sweet potato research, processing, and product development and outreach portfolio. As you will see and hear in this symposium, our scientists are addressing topics and issues related to the sweet potato through creative, state-of-the-art innovation and discovery efforts. In partnerships with other scientists in the land-grant university system and international NGOs, they are directing all that they do towards addressing our stakeholders' needs. I've been looking forward to this symposium and the exchange between presenters, collaborators, industry partners, legislative ag liaisons, and everyone who has taken the time to join us today. So it's my pleasure to be here and welcome everyone. Thank you.
Thank you so much, Dr. Harris. Very, very appropriate comments, and we really appreciate the support of our Southeast area leadership for the sweet potato program, not only in the Carolinas, but all across the country. So we truly appreciate it. Folks, as I said earlier, we don't work in silos. We work in collaboration with a lot of people, and we are not random. We actually are very strategic in terms of what we research about and where that research goes on. And to provide that oversight leadership, we have experts who are called national program leaders at our headquarters in Beltsville, in Maryland, ARS headquarters, national program leaders who oversee all these research projects which go, and I've given you a number, 690 so far research projects carried out all across ARS. Two national program leaders who are very near and dear and close to my heart are the ones who oversee our sweet potato portfolio. One is Dr. Joe Monanenza, and second is Dr. Jean Lester. And I've taken the liberty to request Dr. Jean Lester to please give us an overview of what his and their national program office is all about and how they provide leadership and guidance to us. Dr. Lester, if you're on, please.
Yes, I'm on. Excuse me. It's a pleasure to be here and thank you for inviting me. I'll try not to repeat what Dr. Ellen Harris so eloquently expressed in her presentation, so mine will probably be even a little bit shorter. But in the Agricultural Research Service, as she said, we have a leader, Dr. Shavonda Jacobs-Young, who's on detail at the moment. But we have basically sort of two sides of the house. We have an administrator side of the house, which has an associate administrator, Simon Liu, who oversees operations. And that pretty much oversees Dr. Ellen Harris and the Southeast Area administrative group of people. And then we have the other side of the house, which is the associate administrator for the national programs, overseen here by Dr. Steve Kappas. And within the Office of National Programs, we then have 4 general divisions: natural resources, as you can see, dealing with water, air, and land. And we have animal production, and you can see the listing of the special areas there. And then we have the 2 areas that directly support and are greatly involved with sweet potatoes, that being crop production and protection. Dr. Joe Munyaneza is in that group, and sweet potatoes in that group are addressed by plant genetics and genomics resources, which we'll hear a lot this afternoon, plant diseases to some of that, primarily crop production, crop protection and quarantine at the Charleston lab, and then crop production. In the area of nutrition, food safety, and quality, I'm the national program leader there, and I oversee the product quality and new uses. That title isn't as probably as apparent as it should be, but basically everything after the crop or the animal has been grown, I oversee that research. And so sweet potato processing, sweet potato quality, sweet potato new uses, those are the areas that are of great interest to me. And both Joe in the crop production and protection area and I in the post-harvest quality and new uses greatly support the sweet potato efforts and welcome this symposium this afternoon so that he and I can get a clearer idea or even more ideas on the problems and issues that concern our sweet potato industry in this country, so that as we move forward in the future, hopefully with new additional funding, so that we can hire additional scientists to address these causes, we can continue to answer your problems and your concerns to keep you the world leader that you are and you must always should be. And just an FYI, I'm probably one of your greatest customers, as I eat sweet potatoes likely every single day of the year. Dr. Qureshi.
Thank you so much, Gene. We hope that you will support sweet potato by at least having it once or twice a week on your dinner table. So glad to hear that. I appreciate your comments very much. And I'm sure Joe is also honored. So both you and Joe have been great leader to us. So thank you so much. Folks, continuing right along, moving right along, I just want to say a couple things about who are the participants in this symposium. This symposium is free. This symposium is open to public. And I am so pleased to announce that we had over 160 people register for this symposium. I don't know how many have signed up yet, but they are trickling in. The categories of people who are signed up for this symposium or registered include our House and Senate staffers, legislator, point of contacts for agriculture. We have academia collaborators. We have state and federal governments. We have North Carolina Department of Agriculture. We have regulatory agencies, APHIS and others joining us. But most importantly, folks, the stakeholders, the growers, the industry people, the producers, the people who have boots on the ground to grow sweet potato all across North Carolina and South Carolina and around the globe. So I thank all of you for really taking the time and joining us this afternoon, because we really want to share all the good things which we do, but most importantly, the last session, which our friend Michelle Granger will be hosting to hear from you. So again, here's the theme. Let's go to it. It's 1:16 on my watch, so we are 1 minute late. I'm going to hand it over. I'm going to first close my sharing of this presentation, but then hand over this program to the facilitator, who is Dr. Phil Beetle, who would be taking us from here. Phil?
Thank you, Mukarab. And I'd like to thank everybody for joining us today. And so I'm going to facilitate the breeding and genetics session, and that will be myself speaking and then Dr. Yencho speaking. And I'll try to get us back on course on time here and try not to.
I just went 1 minute over, so.
I'll get us back on schedule.
Thank you.
Okay, so here we go with my talk for today. So I'm going to talk to you about what we are doing with sweet potato breeding and genetics at the Beetle Laboratory. And the primary thrust of our program is for breeding for insect and nematode resistance in sweet potato. And the insect resistance has really been the thrust of ARS since the inception of the sweet potato breeding and genetics over 50 years ago. And the program has been credited with over 75 public releases, with 13 of these being insect resistant. And some of these are highly insect resistant. And we see one of these right here, which is ruddy, the cultivar ruddy. And this is one of the Czech insect susceptible controls. And some of the insects we're primarily dealing with are the wireworm complex, or the WDS complex. And then we have the sweet potato flea beetle. We have sweet potato weevil, and then we have grubs. And so, as was mentioned by Dr. Lester, we're part of National Program 304, and the title of our research project is Basic and Applied Approaches for Pest Management in Vegetable Crops. And I focus primarily on sweet potato and mostly in the development of germplasm that I mentioned is resistant to soil-dwelling pests, and primarily the wireworms, weevils, and grubs and the flea beetle. But we also focus on developing germplasm adapted to low-input, sustainable production systems, and especially with the focus under weed competition. And we're looking at mainly in our project this cycle in the importance of goosegrass competition. But in addition, we are also going to characterize the genetic diversity and population structure of the sweet potato weevil within the US and hopefully worldwide. And as was mentioned earlier, the sweet potato project, we work very closely with other disciplines to facilitate development and release of improved germplasm. And I really enjoy this slide right here that I think gives a great overview of what plant breeding is today, and how you make crosses, and everybody thinks conventional plant breeding is you're just making crosses, you're doing trials and looking to release cultivars or germplasm. But as a research geneticist, and we're trying to also genotype individuals, to phenotype individuals to get at the marker trait associations. And then also on the side of that, bring in mechanistic studies with plant physiologists, molecular biologists, And, working closely with multiple disciplines such as plant pathologists, nematologists, entomologists, food scientists, bioinformaticians, computer scientists, and other breeders and geneticists. And for us, making the crosses, we have to integrate all this information into germplasm development. We start that in a sweet potato polycross nursery, which is just, you have an open pollinated field with up to 25 advanced clones or material that has traits that are of interest, such as insect resistance, nematode resistance, quality traits such as dry matter. And so we start with making these crosses, harvesting the seed, and then in the end of that year, we take our seedlings. In our program, we're running about 10,000 seedlings per year into a field and making a selection based on insect resistance, consistency of shape, consistency of skin color and flesh color. And we are keeping about 1% to 2% of those 10,000 seedlings after the first year. And then that material then in the second year goes into replicated field plots and is further evaluated and eliminated as we find material that is not up to par for releasing for cultivars or advanced germplasm. And so as a geneticist, I mentioned, we're taking all this other information. And one of the recent studies we published was on the genetic diversity and population structure of the USDA sweet potato germplasm collections. And this was an important study with regard that there was a lack of information on the current population structure and genetic diversity within our collection at the vegetable laboratory, but also the USDA sweet potato repository in Griffin, Georgia. And that collection in Griffin, Georgia makes up the backbone of the genetic diversity that's conserved in the US for sweet potato. You also have repositories in Peru, the International Potato Center, and then there are other global repositories that have diversity ready for breeders and other researchers to characterize and to provide useful knowledge with regard to sweet potato. But what we found in this study was that for the good part is we found that material was fairly diverse. We found that of the 60% of material that we found, of the percent that we studied, 40% of that material had mixed ancestry, which is really important because genetic diversity is very critical for maintaining yields, but also for being able to deal with emerging issues such as the guava root knot nematode. And that was a very, very exciting study because within this study, we found 4 different main populations, and these were from various parts of the world. But what I want to focus in is on our North America material. And this is right through here, if you can see my cursor moving, my mouse moving through. And this is this material is from North Carolina and Louisiana. This is the initial populations that started back 50 plus years ago at the US Vegetable Laboratory. And we see these are highly admixed and have diversity, have genetic background made up from everything around the world. This is our current material. And we think this shifting color that you see, which indicates a different genetic background, is from over the 50 plus years of selection for insect resistance. And so knowing this, after I saw these results, I was really, really critical for me to start introgressing more of this material that has been well characterized into those breeding nurseries for maintaining genetic diversity. And I want to mention this line right here is a very critical line. This has resistance to guava root knot nematode. But in addition to that, it makes up a large percentage of the North American background. It was used in initial crosses for all of the public programs within the US. And this is, we think we can trace this back to some of the resistance that we have found within sweet potato germplasm at all the researchers that have screened it. And then additionally, so I mentioned that it's important to know about the phenotypes as well. So again, here's another study that was published recently on the color and dry matter contents of storage roots for the entire sweet potato collection in the Taos in Griffin, Georgia. And this provided comprehensive information for storage route descriptors within this collection that was lacking. There was only, for some of the individuals, there was between 30 to 35% of the data available, and then for others up to 50. And so this is very critical to have this information for designing crosses, but also to use that genetic data that I just showed to actually integrate the phenotype data with the genetic data to find markers that are actually associated with color and dry matter that might allow us to not screen as many individuals, but in the initial stages of selection. And the good thing was the collection, again, is very diverse. We have normally distributed orange flesh types, yellow flesh, and then a cream and white flesh. And then again, dry matter was normally distributed across the two color classes.
2 minutes.
Thank you. And this is the last, I'll speed up. This is something we're really interested in looking at. And we have a postdoc that is going to be starting on this project in about 2 weeks. And it is to speed up our selection process for insects and nematodes. And we see here that we have images that have been characterized by a automated image analyzer. And I've labeled these images, and we've ran these through a artificial neural network to pick up actual spots of damage. And then we've also done the same with nematode infected roots. This is a non-inoculated and an inoculated test, and we can see the difference. And then I'm going to breeze over this to really highlight some of the collaborative work that we're doing with NC State, University of Tennessee, and with Breeding Insight, which is a partnership with Cornell. And I was very lucky been able to select it as a pilot crop in this program. And the goal of this is to take all of this information and to bring together informatics along with the genomics and the phenomic tools to really speed up the breeding process. And so far our efforts we have developed a set of markers for sweet potato and also for sweet potato weevil for characterizing diversity, but also for other aspects. And one thing I'm really excited about with this partnership is the development of a database and barcoding system for data management within the program. Because we're looking at so many numbers, there's so many opportunities for us to propagate ARS into our material, but also we are trying to move away from just paper and pen and really speeding up our time and putting our resources into other efforts. And then I just want to close on, this is really what we're trying to do is bring all of this information. Like, for example, here was two advanced clones of ours at a food science trial with a processor in South Carolina. And then really just bringing all of this information through crosses, marker trait associations to get at a cultivar release, or at least a publicly released clone that is, tolerant or improved for multiple traits that are lacking within sweet potato. And one of our final thrusts has been, we have been breeding for modified plant architecture. This is Beauregard, and this is about 8 weeks after planting. And this is Carolina Bunch, which has been released as a competitive cultivar against weed pressure. And we are looking at this advanced clone right now, which has much better insect resistance than both of these combined. And we're hoping to trial this in the next couple of years. And with that, I'm gonna go ahead and move on to acknowledge all of the collaborators and the various disciplines and participants that have really helped me get to the point that we're at. I mean, I could not do any of this alone. And I think that's what the highlight of this symposium is how collaborative these efforts are. And with that, I'm going to move on to Dr. Yencho, and I'm going to introduce him now.
Thanks, Bill. I'll get queued up here, Phil, if you want to start.
Looks good.
There you go. So you all ready for me?
Yeah, we're ready for you.
Okay, great. All right. Well, thanks, Phil. I think one of the things that I want to say is partly there's going to be a little bit of overlap here between our talks, but partly my goal is our breeding project's goal is to use the diversity and the germplasm that Phil's developing and to help make new varieties out of that. And that's partly how we do a lot of work together. So the first thing I want to say, let me make sure is, breeding new varieties is a rather difficult endeavor in complex sweet potato. And a lot of traits have to come together to make a new variety. And this is just a list of our basic core traits that we're generally evaluating any new variety for. Of course, a couple big things over here, the yield and yield stability is always going to be a core part of our work. But more and more, as the industry has changed and as the industry has grown, I find myself spending more and more time worrying about the chemistry of sweet potato storage roots and the culinary quality of those storage roots too. And that out of necessity has really stimulated a really broad and really deep connection with the food science laboratory here at NC State. Indeed, they're probably one of our largest collaborators in the breeding program these days. And so I want to touch base on a couple of items along those lines. We've been lucky. We've released some fairly successful varieties. Of course, everybody's familiar with Covington. We have 2 new purple lines that we're releasing this year that we're really excited about for the purple flesh table stock specialty market. And we just released a line named Monaco after Dr. Tom Monaco, who actually recruited me as a faculty member and actually paired me up with Ken Pacoda to manage the sweet potato breeding program here at NC State. Monaco's released this year. It's a really nice variety, very tasty variety. Maybe not quite as popular as Covington for a number of reasons at this point, but we think this is actually going to be a good variety for the organic production system. And actually it shows good resistance to waterworm resistance because germplasm that Phil's group developed was used in the parentage of Monaco to provide that insect resistance. So that's a tangible connection to the USDA-ARS effort that we're already benefiting from in the development of new varieties. As many of you know, the growth in the sweet potato industry across the US, but particularly in North Carolina, has been nothing short of phenomenal since about 2005. It really is remarkable. You can see that we released Covington in 2005. Covington's helped for the growth, but it's by no means the only reason for this growth. A lot of factors have come together to help grow our industry. High quality seed, ability of our growers to produce really high quality raw product for a variety of venues, excellent storage. We can store the crop up to and even past the year. New product development that's occurring across a wide range of partners. Exceptional marketing, and the perception of sweet potatoes as a superfood continues to grow, and that's really kind of helped propel the whole industry upward. And then put on top of that the growth in the export markets, and a lot of good things are happening in the sweet potato industry these days in terms of the growth. A lot of times our growers say that a good variety is like the engine. You got to put a good body around that engine to really make it purr. And I think that's probably a good analogy. Covington helps, but you've got to put all the other factors in place to kind of make the industry go places. One of the aspects that have really made the industry grow is sweet potato products are expanding. Many of you know that I work in potatoes also, and I really view potatoes as kind of an emulation of what can happen to sweet potatoes, and indeed that's happening quite fast. A lot of new products are on the market. This is a little bit tongue-in-cheek. Maybe one of my favorite one is the Covington Vodka. It's a very good vodka produced here in Snow Hill, North Carolina. That's just kind of an example of the range of products that are now being created from sweet potatoes, even really good award-winning vodkas. But the thing that has changed my program and my life considerably is really the advent of sweet potato fries and chips and these new processed products in our breeding program. That came about as a result of some big changes in the industry. Most of the major potato suppliers getting involved in sweet potato work too. That started to occur around 2008. And it really has driven some major changes in the program. And it's challenged us as a breeding program to develop new tools to develop new varieties. And I want to touch base on that here really quickly. So I want you to think about the opportunities that are provided in a cross like this. This is a cross between an orange flesh sweet potato and a purple flesh sweet potato. And this panel here shows 20 siblings out of that cross. And from a genetic point of view, I call this segregation, all right? So each one of these discs here represents a potential new variety, all right? They're a distinct clone out of a cross between a purple and an orange sweet potato. I want you to think about the functional food products that might be created in this in terms of the anthocyanin levels, which causes the purple, beta carotenes, the orange, phenolics, lower glycemic index in here. You know, this is probably why so many people are consuming sweet potatoes on a weekly and in some cases daily basis. Thank you. But also think about the new flavors, the colors, the different sugar, starch, and texture profiles that exist in here, and the potential to reduce acrylamide levels during the frying process. So my point here is there's a huge number of opportunities in this cross, but the thing is, the challenge is we have, how do we decide what that we want to develop and what tools do we use to help us get there when we're creating a new product or a new variety from which we want to derive new products. So we've done a lot of work with the Food Science and Market Quality Research Unit over in food science at NC State over the years, and Den Truong's on the call, and I think he's going to talk a little later. We've done a phenomenal amount of work with that team over there. They're really stellar, and we've learned a lot about the chemistry of storage roots and sweet potatoes. To a certain extent, we can do it by, I'll call it brute force. We bake, we fry, well, we chip and we fry samples in large numbers. And by that, I'm talking about, you know, dozens of clones, if not tens of dozens of clones, sometimes hundreds of clones. We'll screen through ones to find the one that might have the brightest chip color out of, say, this is what it used to look like, and the brightest chip color. And same here. So one of the tools that we've used is NIRS, near infrared reflectance spectroscopy. And now we can sample thousands of samples a year. And off of one screen, we get a measure of moisture, dry matter, all these sugars, starch content, amylose, asparagine. So this is a new tool that we're using to help us understand storage root chemistry in sweet potato. And it's been a very, very powerful tool. Another tool that we use is our Exeter Sorter, which is down at the Horticultural Crops Research Station in Clinton. Industry and commodity groups helped us to purchase this. It's state of the art, and it emulates what's going on in industry right now with some of our major packers. So we can pack out materials and sort of emulate what's going on in industry with our single line Exeter Sorter at Clinton. All right, Phil alluded to this, and there's now a new advent of genomic tool availability to use to facilitate in sweet potato breeding. And really, a lot of our major advances came about as a result of a rather large-scale investment by the Bill and Melinda Gates Foundation for improving sweet potato production in sub-Saharan Africa. It's a long story to go through. I was tapped to lead, NC State was tapped to lead what we call the Genomic Tools for Sweet Potato Improvement Project. And I want to share with you some of the outcomes of this project, which is now being followed on by a new project called Sweet Gains, which NC State is also very deeply involved in. It's led by the International Potato Center down in Lima, Peru, and it has substations all around the continent of Africa that are working on new tools for sweet potato improvement in Africa. But these tools also benefit us here at home.
Greg, we're out of time. That's the 10 minutes.
Oh, sorry. How do you want me to just stop right here?
I think we can maybe discuss this if there are no questions that come in. You can.
Let me wrap up one thing here, and I'll go to my last slide.
I do not have any questions in the chat, so Dr. Yancho, you could wrap up.
Fair enough. Sorry about that. Okay, so Phil kind of talked about this a little way earlier before, but we're now what I would say in an era of sweet potato breeding, in the era of genomics. And the things that I think Phil mentioned, I really want to agree with him, and I want to really emphasize it even more. Breeding today takes multidisciplinary teamwork in which to build a new variety. It takes advanced training. It takes a reference genome, which we now have for sweet potato. It takes advanced genotyping technologies, which we now have. And because sweet potato is so complex and it's very different from most other crops being a complex polyploid, it takes new analytical means to measure traits and to do marker trait associations. We have the capability to develop very high quality breeding populations. And if you're dealing with all of this information over here, you really need to have a robust database and electronic data capturing methodologies. And so we have all of this in place right now. We have the teams in place. The trick now is to make this all purr together. And I think I'll conclude with my last step is to do this, you need lots of partners. And I just will finish on this one. This is a list of our partners, major partners to kind of help us do and engage at this fairly high level of research to make new varieties of sweet potato. Sorry for going over on that, Phil. I thought I was right on time, but I guess I missed it.
No worries. And thank you for the, it was really good talk, and I really agree with you, the emphasis of this teamwork and having the ability and the advanced training. And do we have any questions out there from any participants? Because I do not see any in the chat as well. We have 3 minutes until we have a break session, but I would mention the new release that you have for the organic market. We have worked with that as well and working on a manuscript that has performed well under plastic culture as well, and has performed well yield-wise as well.
Yeah, the other thing I would add is that also fits into the weed management too, because it's like you mentioned, it's that bunch type that helps to suppress weeds early on in the season. And that fits in well in an organic situation or a conventional situation.
Exactly. I guess we can go ahead. If there's no questions from the audience, we can go ahead and take our break a little bit early. And
if I could make a comment, Phil, this is Mokura.
Hold on. We just had a question come in from Jan Lowe, and what is the diversity of purpose types in the USDA collection? Okay, there you go. I was wondering what purpose was. So purple types. So the diversity of the purple types, my recollection, there are very few purple types in the collection. And you've caught me off guard on exactly how many are there, but I want to say there are 10 or less purple sweet potato types in the collection, and we did identify through some of this work some mislabeling within the collection, and which is not uncommon in clonally maintained collections. I think our rate of error was very similar to some of the rates that have been published elsewhere. We have another question: is sweet potato breeding focused primarily on industrial and field relevant characteristics, or is it healthfulness and bioactivity also considered in modifying germplasms? Craig, since you were talking about your focusing really on the chemistries, I'll let you answer.
I can tackle that one. More and more, and as we become more sophisticated, we're more interested in the healthful aspects and the bioactivity aspects. But that's not to say the productivity issues still need to be there. So what we need to do is to keep productivity, but I think we're learning more and more all the time about the nutritional and particularly the bioactive nutritional aspects. And I think that's very perceptive. But that's a really important aspect of the breeding program. And as we learn more in the food science side of things, there's opportunities that are opened up along those avenues to kind of keep the agronomic quality, but enhance the nutritional quality.
Well, I don't see any other questions coming in, so.
I'll tell you, I can go back to Jan's question on the purple sweet potatoes. And this is a shout out for the International Potato Center. The germplasm base of the purple genotypes is actually rather low. And a lot of the purple materials that actually started here were derived from some purple polycross nurseries that were done by the breeder in Lima right before Wolfgang Grunberg started the job. I'm drawing a name blank on his name right now, Jan, but you know who I'm talking about. And that was True Seed that was imported in here of purple parentage, and a couple of small purples were floating around. So I would speculate that it's very, very narrow.
There's one question coming in for me about USDA's role in releasing Finnish sweet potato varieties. I think on that side, I think we really have put together some partnerships. Like Craig and I have a partnership where we are sending germplasm for joint selection and evaluation and potential release. And I think that's more our role is really is working together with the other public parties to release improved material that has the, as Craig was mentioning, the release for organic with some of the background from our program, but then also direct material from our program going to North Carolina and Mississippi and Louisiana to actually have that material evaluated under those conditions in the initial selection stage is critical, I think, as well. So that's kind of what I see as our part in releasing finished varieties rather than in a improved genotype.
Excellent, Phil. I think we can possibly stop here if
Makarov,
Yeah.
This is Gene Lester. I'd like to make one statement. I heard
Yes.
earlier this week that the Food and Drug Administration is now very concerned about chemicals, specifically heavy metals and pesticides in foods, in reducing the exposure primarily to babies and children. And where they're coming from, they have now moved from parts per million to parts per billion, simply because they now have the instrumentation to be able to do so. We need to be aware of this because we may be seeing we have a problem where there's currently not a problem existing today. So our continued collaboration with the Food Science and the Market Quality Laboratory to hopefully beef them up to help in this situation is going to be critical to the sweet potato industry, especially for baby foods. I just want to give you a heads up.
Thank you, Dr. Lester. Very timely, and believe me or not, Phil Wedel and I have been exchanging a lot of emails and a lot of brainstorming around this area. We are really gearing up to request some additional funding from some other federal competitive agencies to make sure that we have the tools and resources to do it. But absolutely, you're right. This has to be the next thing which we need to tackle. And I think our chemistry labs ought to step up and do this. So thank you so much for pointing this out. Folks, I think, Phil, if there's no other question,
there are a few more that came in, but we're into our break is about over.
But I would say so. I would say this, that every question which has come in and we are not able to answer, we will get these answers and post these answers along with the recording, and also email all the responses which we get to all the participants. So thank you so much for a great session, Phil. Thank you, Dr. Yanko, for always it's a great pleasure to hear from you. So let's take a couple 3 minutes break and reconvene at 1:50. That gives us only 2 minutes. So I'll ask Dr. Suzanne Johanningsmeyer to start getting her session presentations ready. So we'll see you in a minute. Don't go away. We'll be right back. Dr. Johanningsmeyer, seems like you're ready.
Yes, as soon as I'm ready. Would you like me to start?
Just one. All right. We are back, folks. You are tuned to Goodness Grows in North Carolina, Sweet Potato Symposium, hosted collaboratively by ARS North Carolina and South Carolina Labs, as well as our wonderful stakeholder partners, North Carolina Sweet Potato Commission. And thank you, Michelle Granger, for being our partner, and all the stakeholders who are attending. I know a lot of people still are signing in. A lot of our legislative agriculture point of contacts are signing in. I've been getting notes, so thank you. You're not late, even if you're joining right now. We are into the second session now, Goodness Grows in the Carolinas. Session 2 would focus on our food science expertise. As you heard previously, a lot of geneticists and breeders depend on the food science aspect of all the wonderful varieties and cultivars they develop. Dr. Suzanne Johanningsmeyer is a food scientist in ARS. So Dr. Johanningsmeyer, it's up to you now. Go ahead, your session.
Okay, great. Thank you. Good day, everyone. And welcome to session 2 by the Food Science Market Quality and Handling Research Unit in Raleigh, North Carolina. We have 3 presentations for you, and I want to invite you now, as you're listening to the presentations, please feel free to type your questions and comments in the chat box, and we'll have a short Q&A at the end of the 3 presentations. So with that, it's a great pleasure to welcome our first speaker, Dr. Van Den Trong. He's our former research leader and led the sweet potato research program in our unit for 16 years until his retirement in January of 2019. So it's a great honor to have him back here with us. And one second, there we go. And to share with you historical perspectives on innovations in sweet potato research and technology transfer. Ready for you, Dan.
Thank you, Dr. Johanningsmeyer. And I would like to thank the organizing committee for having me to participate in this special sweet potato symposium. It is great to virtually meet all of the friends of sweet potatoes. So as mentioned, I'm going to talk about the historical perspective in relation to innovations in sweet potato research and technology transfer. Next, please. I think Suzanne, could you? Yeah, okay. So to mention very briefly about the history of our sweet potato research program at USDA-ARS in Raleigh. In 1935, the USDA-ARS fermentation labs started, and almost 30 years later, the carotenoid biosynthesis project was initiated. And 5 years later, in 1969, it was converted, redirected to the sweet potato research to meet the needs of the sweet potato industries. So the two scientists, Dr. Albert Purcell and Dr. William Wanter, work on the sweet potato chemistry, post-harvest handling, storage, biochemistry, and also processing of the sweet potato into dehydrated flakes, purees, the frozen and fried products. And following these two great scientists who left behind the big shoes for filling in, I joined the unit in 2003. Next, please. And to carry on the mission of this the sweet potato research program, we conduct the basic and applied research on chemistry, processing, and functional property of sweet potato that contribute to increased consumption of this highly nutritious vegetable. So we focus on chemistry, processing technology, and product quality. So our goal is to increase the consumption of the sweet potato. But if we look at the sweet potato farm, the main markets of the sweet potato is the good side and good shape sweet potato roots, or in other words, that the #1 the sweet potato root for the stock market. And about 30 to 40% of the jumbos or the small size sweet potato are not utilized. And on the other hand, the food industry can incorporate the nutritious fresh sweet potato into various products to increase the consumption of the sweet potato. So there's really a need of developing the processing technology to convert this highly nutritious sweet potato into the functional food ingredients that can be readily utilized by the food industry for a food product, and with that we can increase the consumption. So in 2008, as Dr. Craig Ciancio mentioned, that there was also really a demand on working on the sweet potato varieties for fried chips and French fries, and that would also eventually increase consumption of the sweet potato. So in my 16 years of doing the research on sweet potato at the USDA-ARS in Raleigh, we focus on the 3 projects. Next, please. The first one is the aseptic sweet potato purees. Sweet potato need, regardless of the size, it can be converted into the puree that can be processed in such a way that it could have the high nutrient retention, shelf stable, and it's easy for the food processor or the pet food industry to incorporate in their products. So we team up with Dr. Joseph Simonovic, working on the microwave heating at that time. And after 2 or 3 years of working on the project in the pilot plant, we came up with the very good process of processing sweet potato into the aseptic packages, and as shown in this photo. And we presented the technology to the sweet potato, the North Carolina Sweet Potato Commission, and farmers and also investors really interested in seeing the technology, and they brought the sweet potato into the pilot plant, and you see that they really want to see how it works. And after a day of working together in the pilot plant, you can see that the team was very happy to see the finished product, that is the aseptic sweet potato with a very good color, high quality. So with that, the big event next in 2008, that's the inauguration of the YAMCO in Snow Hill. And we see here in the picture, the Agriculture Commissioner, Steve Troxler, gave the inaugural address at the celebration, at the inauguration. So, and the team also received the USDA Technology Transfer Award and also the IFT Food Technology Industrial Achievement Award in 2009. With that, next. And in 2017, in collaboration with Dr. Tawanda Businji, and also Joseph Simunovic, and with the funding from the USDA-ARS, we hosted also sweet potato meeting, talking about the possibility of introducing the sweet potato puree processing to the CIP SIP program in Africa. And just only, and then the company Sinovatec, located here, right here in Raleigh.
2 minutes.
Yes, Sinovatec LLC, yeah, work on the development of the sweet potato processing puree facility. One was established in Kenya, and we just learned lately that the other one will be established in South Africa. So, and then Dr. Simunovic and Muzingi may have also additional information about that. And then those are the things that is quite exciting for the sweet potato puree processing technology. Next. Next, please. The second approach we have been working on was instead of grinding or cooking sweet potato into the puree and process the whole thing, we can fractionate the sweet potato roots into different components. One is the juice, and then we extract the juice, and then we also have the residue to dry it up into the flakes, or we can separate the starch component. So we can have not only one product, but we can have on the 3 products. And for the sweet potato, for the starch, we can also hydrolyze it and put it back to the juice, so to increasing the concentration of the sugar level in the product. Because in the US, the sweet potato, the starch is quite cheap, and then there's also difficult for introducing the sweet potato starch in the market. At that time, in 2011, John Kimber at the Sweet Potato Commission also worked on the dehydration project, and then we collaborate with Dr. Mike Boyett in Ag Engineering, and we had also the demonstration of the dehydrated sweet potato in the farm in one day. One of the things is drying sweet potato, because of a high level of moisture, it takes a longer time. So the group thought of kind of reducing the moisture content of the raw material by grinding and pressing or to reduce the moisture content, so we can shorten the drying time. So with that, John Kimber worked on with the group of investors. And this is also another big event, the groundbreaking ceremony of the sweet potato processing facility in Nashville in 2014. And on the research team joined this exciting date in Nashville of the groundbreaking for the company. And I think the company started to operate a year later in 2015.
We're out of time, Dr. Trong.
Yes.
So I will take you to the end of your slideshow to wrap up.
Yeah, okay, next one. Yeah, and then the third one here is, okay.
We're out of time, I'm sorry.
All right, yeah, so.
I'll take you here.
Right, yeah, anyway, we would like to mention about could you go back to the previous slide just 1 second? Yeah, I think here is also, aside from the collaboration with food science at NC State, biology and ag engineering, and then the sweet potato breeding program, as Greg already mentioned, we also collaborated with USDA-ARS in Charleston. The previous year, we got also root sample for evaluating, but because of the weevil problem in the past few years, lately, I think we could not really receive the sweet potato root from Charleston, and then it goes through the sweet potato breeding program at NC State. We collaborate with the AS in New Orleans for sweet potato and rice product for the gluten-free product, and also the USDA in California for extrusion, and also in Madison for the acrylamide. And as mentioned, we collaborate also with International Potato Center. And we work very closely with the sweet potato industry, US Sweet Potato Council, and Sweet Potato Commission, and sweet potato farmers and food company, and aiming to increase the consumption of the sweet potato. So the key thing is, I would like to mention, is collaborations is the key to innovations in research and technology transfer. Next.
Thank you, Dr. Trong.
Okay.
We can please put your questions in the chat, and we can continue the conversation in the Q&A.
Okay, great. Yeah, with just that one.
Our next talk is, I'll be giving the next talk on breeding tasty sweet potatoes, influence of storage root biochemistry on sweet potato quality and consumer preference. I'm a research food technologist with the USDA, and my program is focused on developing science-based technologies for preservation of fruits and vegetables to enable the sustainable production of high-quality, health-promoting consumer products. Of particular interest is the sweet potato for a variety of reasons, which you've already started to hear about here this morning. So one thing is that the dietary guidelines for Americans, the most recent version came out last year, and they continue to encourage us to increase our intake of fruits and vegetables. In particular, at least 90% of the US population is not consuming the recommended intake of red and orange vegetables. So among the fruits and vegetables that could help to address this need, sweet potato actually produces more nutrient-rich biomass than any other food crop worldwide per land usage. The nutritional value of especially the orange flesh sweet potato is notable and is being promoted worldwide to combat vitamin A deficiency, as well as the many other nutrients that it contains that can reduce the risk of chronic diseases. So not, all of that, the nutritional value, of course, is a strong point for sweet potato, but I think this high biodiversity in the germplasm is especially exciting, and you already, Craig set me up well for this. So it's pretty exciting, and I think that the vision for the future is that when you walk into any mainstream grocery store, you'll see this whole variety of sweet potatoes to meet your culinary pleasures of different flesh colors, flavors, and textures, just like we currently go into the grocery store now and we see a whole row of apples of all sorts of types for different consumer markets. And this also opens the door for development of novel products, processed products as well that are more convenient. I did want to note that USDA has been supporting sweet potato growers for more than 100 years. And I found this really interesting that very early on, the importance of the culinary quality of sweet potato to improving the consumption of this crop was noted here in this 1901 publication on how to cook sweet potatoes for full flavor development. And so it's through this combined effort that we really make these advances. So the thesis here is that increased utilization and consumption of sweet potato can be achieved through integrated advances in breeding, crop management, and food science, and that food science has an important role to play in that, especially with regard to eating quality and developing varieties and processed products that meet consumer preferences. And together with these integrated efforts and collaborative efforts with breeding programs at NC State and the International Potato Center, we can achieve this goal of increasing utilization and consumption of sweet potato. And so in our lab, we use a combined approach with using advanced analytical technologies and developing new methods for being able to look at the underlying chemical composition of sweet potato and sweet potato products in combination with product quality tests that are more empirical in nature, such as instrumental color and texture measurements, and combining all of this information along with sensory analysis so that we understand the human and consumer perception of these products. And so I'm going to just take you through quickly a few projects that where we've incorporated sensory analysis into further development of sweet potato or sweet potato varieties. So one of the things to note is that sweet potato takes a long time. It can take a long time to cook to get that full flavor development. And so that can be a deterrent for consumers to consume sweet potato because of the extra preparation and time that's needed. So early on, we did a study looking at a more convenient product for consumers, which were fresh cut sweet potato dices in microwavable pouches. And we optimized our microwave cooking protocol in order to get the right texture and flavor into these, and then evaluated them using a difference test compared to freshly prepared samples. And the goal here was to look at different washing regimes that are used for fresh cut sweet potato products and how they helped with the microbial stability and then also influenced quality of the finished product. Key take homes were that we did note an off aroma in the sweet potato with chlorine rinse treatments that are commonly used in fresh cut operations. However, there were alternative sanitizers that were very effective in retaining the appearance and flavor similar to a freshly prepared sample up to 14 days in storage. We've also partnered with the Department of Horticulture to look at different cultivars and how they compared for oven baking or microwaving as whole roots. And the key take homes here were that once again, even after optimizing the microwave cooking protocol, the oven baked sweet potatoes were more well liked for not only their flavor, but also their color and their texture. And we found out by using just about right scales with our consumer tests that this was due to a lack of moistness, flavor, and sweetness in the microwave sweet potatoes. It was also interesting that there was a clear preference for the deeper orange color of the sweet potato. Another way that these type of consumer preferences can be evaluated and used to compare different sweet potato varieties is shown here, called a penalty analysis. And what we're showing is that this is one variety, and the percentage of consumers that said it was not firm enough or not sweet enough was relatively high. And they also had a decreased liking for this variety due to these factors, whereas this other variety doesn't have any attributes falling in this quadrant, so means that it was pretty spot on and it had a high overall liking score. And so in looking at, okay, so now how do we take this consumer information and put that in the context of a breeding program, where, as Craig mentioned, you may have, hundreds of cultivars that you want to evaluate. In order to do that, we need to develop a objective way of understanding the quality traits of sweet potatoes that accurately reflects our human sensory experience. And to do this, we use trained descriptive sensory analysis panels, and we started the development of an international sensory lexicon for sweet potato with our partners in Uganda at the National Agricultural Research Organization, and this work is ongoing. And so just briefly to give you an idea of what this looks like, we, in a very intensive work session with about 15 tasters, we developed 36 descriptive terms for the sensory quality traits of a variety of sweet potatoes from their breeding program. And we found that these were organized into different types of traits of aroma, appearance, flavor, and then many texture traits that are very complex and more difficult to measure. So we actually had to develop very specific methods for how you evaluate these different traits so that they can be evaluated consistently and objectively. And this is, we're getting away from how much someone likes it and using the human senses as an instrument to describe the overall characteristics of a sweet potato. Similarly, in an earlier study we used a descriptive sensory analysis panel to look at the texture quality traits of sweet potato French fries. And for these we see we had 16 different cultivars that varied in dry matter content. And wherever you see a blue dot, that is a different cultivar, and they're arranged on this map of flavor attribute or texture attributes, excuse me, according to their texture characteristics. So for example, Covington, Evangelina Beauregard, and a couple of experimental cultivars are all grouping in this quadrant where things reflecting a more smooth and moist inner texture of the fry and inner texture that kind of sticks to itself while you're chewing it. That's what this cohesiveness of mass represents. Whereas as you move to the left on this map, you see increase in hardness, crispness, and exterior roughness, more similar to the coated commercial sweet potato French fry that we were using as a reference. And one of the key distinguishing features between white potato French fries and sweet potato French fries is this characteristic of denseness. And that is the kind of how compact the middle of the fry is versus the very light, airy fry that you get with a white potato. And this is something that couldn't be predicted based on dry matter content alone. And so it requires further looking into the underlying chemistry of sweet potato to understand other textural traits that are important to consumers.
Time is up, Suzanne. 10 minutes up.
Matt was supposed to give me a 2-minute warning. Okay.
He did.
Maybe I wasn't loud enough. I'm sorry.
Sorry, Matt, I didn't hear you. Okay, so
I think I'm seeing a trend here.
wrapping up then, we're continuing our work to look more deeply into the underlying chemistry that will affect these important texture properties and sweetness that really drives consumer preferences. And of course, acknowledgements, I think these slides will be shared. So I'm going to wrap up with that. Encourage you to keep chatting in your questions and comments, and then introduce our final speaker in this session, Dr. Matthew Allen. He earned his PhD and did his first postdoctoral training at Purdue University in food science with a specialization in carbohydrate chemistry. And he joined our research unit in 2020 as a research associate. And he will be wrapping us up talking about strengthening the sweet potato industry through innovations in processing and variety development.
Thanks, Suzanne, and sorry that I didn't say it loud enough. But anyways, yeah, like Suzanne talked about, I will be talking about what she introduced. So the aim of the Sweet Potato Research Program in the Food Science Market Quality and Handling Research Unit based in Raleigh, North Carolina, is ultimately to provide value to the US sweet potato industry. And if you look at the bottom right, that's not a made up graph. That's the actual real graph of the US sweet potato industry growing in the last 20 years. It's almost comical to me to just see a commodity grow that much. So it's exciting times to be doing research or being a part of such a booming industry. I like to think of that. The Carolinas is kind of the Silicon Valley of the sweet potato world. So we are the headquarters. I'd like to think we're the headquarters of the world for sweet potato research and production. But like I said, the goal of this whole unit is to provide value, or research that provides value to the US sweet potato industry. And Dr. Van Den Trong, he was a giant in this research, and he greatly contributed to the sweet potato industry. And we hope to continue doing such valuable research in this research unit to continue this upward trend of the US sweet potato market growth. So this is just a really broad overview of the type of research schematics that we go through in our research unit. So usually we're addressed with a problem, a concern from the sweet potato industry, or collaborations with sweet potato breeders. We're trying to compare varieties. And we process those products into anything that's available on the market. We can do chips, fries, purees, flakes, baked. We can process them into any product that's available. And then we compare those different varieties or treatments using sensory analysis. And fortunately, we have an in-house expert, Dr. Suzanne Johanningsmeyer, who is an expert in sensory evaluation. So we can have trained panels or untrained panels to then compare these different varieties or treatments and see what's different about them to the consumer. Is it good? Is it desirable? Or is it bad? And what is different? And we could also do some objective measurements using like the maximum force to break a chip or the viscosity of a puree. So we can also do those objective measurements as well. So we can get a good understanding of the different products. But then the unique thing is we can also dive deeper into the chemistries. This is where I get really excited. It's all exciting, but this is the stuff I really love, is the chemistry. What is making that sweet potato product A different from sweet potato product B? And so we can jump into small molecules, the aroma compounds. We could look at the composition, how much sugars, the different types of sugars are in there. We also can look at the pectin, starch, and those compositions and dive deeper into their molecular structures and see how those different compositions and molecular structures can affect that sweet potato property. So we can have a variable, a treatment, or differences in varieties, and then we can attribute that or those differences to those sweet potato products. And with this type of research, then we can deliver it to the breeders and the growers and the processors to then give you the foundation to make informed decisions, as well as continue to develop this US sweet potato industry for the bottom right graph, continue growing this US sweet potato industry by improving the products that we have. Next slide, please. Oh, the animation. And of course, this is not just a symposium to talk at you. This is a symposium for us to listen to. So all these research that has been done over the decades, and the valuable research is not started off our own creative ideas. It started with people like you giving us feedback of what are the needs of the sweet potato industry. So we value your feedback, and I please reach out to me, Suzanne, Dr. Kresge, or anyone in the symposium. My contact information will be in the bottom left of all these slides. Next slide. So I just want to touch a little bit about what has been done really recently, as well as what we're doing right now in our research unit. So recently, Suzanne touched on this. There was a study done out of our unit that associated sweet potato French fry textures to their compositions. And there's a lot of strong correlations there where they found that, for example, higher sugar contents were associated with more moist and softer French fries, and higher starch were associated with crispier fries. But it was not, there were some uncharacterized textures, like Suzanne said. The denseness is still kind of was out there. It was hard to pinpoint what was causing that. So we looked a little bit deeper into the starch. And so we isolated the starch and characterized the starch. And on the right is an example of sweet potato starches that were isolated from these different sweet potatoes. And then these are their thickening profiles. And you see the lines that represent these sweet potato varieties, the red line and the orange line. Those aren't orange per se, orange varieties, but those are, well, they could be. Those had higher thickening power. And the blue and the purple lines represent starch that came from sweet potato varieties that had less thickening power. And what we saw was that the sweet potatoes that had starch that had very high viscous starch tended to be in fries that were perceived as dense, and fries that had less thick starch tended to be less dense. And our thought was, since the starch is inside the cells, when you heat it and you cook it, maybe within those cells, you're creating a high local viscosity. And so when the consumer is eating it, it's perceived as dense. So that then generated this general scientific hypothesis. It's not just composition, it's also component molecular structures that affect the textures of sweet potato products. Next slide.
You have about 1 minute.
Okay, well, all right, next slide. Sweet potato, so what biopolymer, so the next thing is what we're doing right now is what biopolymers really influence the sweet potato textures the greatest? Is it the starch? Is it the pectin, the structural proteins? Is it cellulose, hemicellulose? So we have all these biopolymers that are in theory, affecting the sweet potato texture in chips, the baked potato, the fries, the purees, but which one's really driving the textural differences? And once we identify it, for example, let's say the pectin is really driving textural differences, what molecular differences between the pectins are driving it? The branching, the chemical substitution, diving deeper into the nitty gritty will then help us do some targeted processing and breeding approaches. Next slide. So once we understand how these components and their molecular structures affect sweet potato textures, this will lead to focused processing technologies and more targeted breeding. So for example, if we think it's the pectin really driving these textural differences, we can do processing treatments. So we know there's temperatures that degrade like pectin, and there's temperatures that promote pectin rigidity. So we can have a targeted approach in how we process our products to promote or degrade a certain component. Or we could use enzymes or processing aids that will be very specific towards certain polymers, as well as variety development. If we want higher composition of a certain polymer, more starch, more pectin, more structural proteins, they can breed and target for those types of constituents, as well as looking at their molecular structures. Like there's well-expressed pathways that show certain enzymes will affect the molecular component structures of these biopolymers. So we can have targeted breeding efforts as well as targeting processing for tailoring essentially our processed sweet potatoes or our table stock sweet potatoes to meet the consumer needs and demands. So we can now broaden and basically customize our sweet potato the way we want. Last slide. And so again, just reiterating, we need to hear from you. So feel free to reach out to us anytime, but also today, we would love to hear from you. We have collaborations with breeders, growers, and processors. So there's no one that's off limits to our collaborations or discussions. And for the ultimate goal, obviously, is we want to help you all in growing the US sweet potato industry. So that's what those blank lines are on the bottom. Those are the things that hopefully we discuss today or in the upcoming months. Thank you.
Thank you, Matt. Great job. And that takes us to, I think we have a few minutes for Q&A. So Dr. Qureshi, do you have some questions for us?
In fact, there are several questions. Great job, folks, in this session. I'll read some of these questions to you, and I apologize if I'm being selective. As I said, there would be a lot of answers posted. One question, are there any regulations being enforced on acrylamide levels in the US? How does the problem in sweet potato compare to potato? I know it's a regulatory question and we are not a regulatory agency, but anybody who could provide some insight in it?
I can provide a little insight there.
Sure.
So there's something called Proposition 65 in California, and they've set some guidance on acrylamide levels that you need to be below, or you have to label it on the products. And so I think that level has been kind of used as a target by the potato industry, and could also be used as a target by the sweet potato industry as a level to be under. The levels that are set in the European Union are actually a little bit higher than that. So I would say there's some guidance out there, but yeah, I can't get into the regulatory side. So there's some guidance that can be used as targets for breeding, And we didn't have time to talk about the acrylamide project, but we do see evidence there that the compositions vary in the sweet potato varieties. And so variety selection could be used as a method for mitigating this so that it is not a problem in fried sweet potato products.
Great, thank you. Another question, if you have time. For food science, there is growing interest from pet food industry and for livestock feed. Is USDA responding to this need? I guess I can share a bit. Yes, you know, for example, sweet potato, as an example, there's a lot of waste being generated when sweet potatoes are being processed, I guess. We are actually carrying out a study, which is feeding trial, using orange flesh, sweet potato waste in formulating diet for chickens and seeing how does it impact food digestibility, how does it affect, let me turn my video on, how does it affect performance of chickens, but more so, are the color or pigments transferred to the yolk to make the egg yolk a little more brighter, a little more consumer accepted. That's one example, but Suzanne, feel free to jump in.
I don't have any other information. Matt, do you know anything on that, or?
No, I know there's a lot of dog treats being made out of dried sweet potatoes, but I don't know what type of research is being led that way.
Yeah, as a breeder, I would say I don't envision that being a breeding goal. It would be, you know, can you find a way to add value to the waste stream coming off of, you know, the higher number one and the export stream? That would probably be my value proposition on that. But there's you know, you're finding sweet potato off products in all kinds of food now. I mean, my dog actually, you know, uses a pet food that has sweet potato products in it. So it's great. A great way to add value to the chain.
Yeah, I think
Thank you.
the pet food industry has been using a lot of sweet potato for their, you know, new food product, new pet food products. So actually, in the previous year, we imported a lot of dehydrated sweet potato from China for the pet food industry. And that's why many companies came to us and then asking for, could we produce dehydrated sweet potato for the pet food industry so that we will not import from other countries. So that is how, you know, we did separate project of the dehydration of sweet potato. I think for the pet food industry, the quality of the product is also as good as a requirement for, you know, the human food.
Sure.
So, and for the waste treatment, I think most of the processing company, they do not really have the way how to recover, you know, the waste out of the processing operation. It's mainly for the animal feed. So, and in the factory like Yamco, we have also a lot of sweet potato puree, that was not really very good quality. And I think they initiated with starting fermentation and then make it an alcohol, all the stuff, you know. So, and that is where we initiated the idea of having the Covington Vodka, you know, so.
which Craig and others enjoy.
Right. Yeah.
One question which might be of interest to all of us, what is the capacity of North Carolina to continue to support sweet potato industry growth? I know it's a very broad term, capacity. What do you mean by capacity? Is it infrastructure, personnel, resources, intellect, industry, partnership, all of the above, I guess? Suzanne?
I can comment on our scientific capabilities, and then I'm going to shift it back to our leadership on the other things that you mentioned. So on the scientific side, we have strong capabilities in analytical chemistry for profiling the composition of small molecules, and we're continuing to make advancements in those areas. And we are building our internal sensory capabilities as well as part of our in-house projects under NP 306. As well as we have our research associate, Dr. Matt Allen. He's here with us on a 2-year appointment. So we have about another year with him. And so I think in terms of the science, we have all of the right things lining up to continue to support the sweet potato industry and propel the growth. So now I'm going to divert that question back to our leadership that have on the other administrative aspects of that.
I think you tackled it real well, Suzanne. Yeah, you know, there are gaps. There are gaps in every program, and our job and leadership job is really to ensure that the resources and the infrastructure is there for us to continue to support. All the success which we have achieved so far has not been with nothing. I mean, you know, really, North Carolina has been a key supporter. ARS has been a tremendous supporter of USDA, of sweet potato industry in the Carolinas and across the country, across the world. Yes, there are gaps, and we are working towards ensuring that those gaps are met. So thank you for the question.
And might I just add that our capacity is very much dependent upon the symbiotic relationship that our program has with our stakeholders. And our role is to keep you informed of what we are doing, to collaborate with you to meet your needs. And our hope is that you in turn will advocate for us in those venues where it's important that resources be added to our programs.
I could not have said it better, Dr. Harris, than you did. So thank you. We appreciate your continued support, but great message. We cannot afford to work in silos. It has to be collaboration, and our stakeholders ought to be huge collaborators for us, because what we do at the end of the day is to benefit the common man on the street. And here is sweet potato commodities. Thank you, Dr. Harris. We truly appreciate the comment.
Yeah, Mukarab, if I could jump in real quick on part of my understanding of the question was, is the industry poised for continued growth? And is North Carolina, a state that could continue on this trajectory? And Michelle, I see you're on the call too, and perhaps later on we'll discuss that a little bit deeper. But my own personal feeling, you know, knowing the industry fairly well and you know, getting out and traveling around quite a bit, is that there's avenues for considerably more growth in our industry. And I would not be surprised if, you know, we're at just shy of, we're bouncing around about 100,000 acres right now. I would not be surprised if in another 5 to 10 years that we're north of 150,000 acres or so. All it would take is substantial investment by one of the major processors to put a plant here, our export markets to continue to grow, our deep water ports expand. So we have the land, and I think that, I forget who said it here, but it might have been Matt, that we're really a Silicon Valley of sweet potato technology and scientific knowledge. And you couple that with the plant sciences initiative that's coming out of the university right now, and just being an RTP in this area, we have world-class sweet potato expertise across the spectrum at a scientific level, but also at a grower level too. So I think we're well poised to continue growing and to grow sustainably over the long term, as long as we kind of, you know, pool our resources and support each other. That's my own personal feeling.
Professor Yancho, absolutely. That's why you are a distinguished professor, to keep us at this very broad visionary model that we all strive towards for. Thank you so much. Folks, this has been a great session. Let's give a big virtual hand to Dr. Suzanne Johanningsmeyer and the food science session, folks. Thank you. Okay. Okay, I think we have eaten into our break. Actually, we have gotten into the next session already. So maybe we can take, let's stop sharing the previous session slides. And I would ask Dr. Levy-Williams to please be ready to take over the session 3, which are the challenges. So Dr. Williams, if you're ready, stage is yours. And please, 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 as you have been, keep posting your questions and comments on the chat box. They are absolutely phenomenal questions. Very, very relevant to the topic we are addressing today. So thank you. Keep doing that, and I'll keep scanning and screening those questions and try to answer them as best as we can. Thank you.
Good afternoon, everyone. My name is Livy Williams. I'm a research entomologist at the Vegetable Lab in Charleston. And Will Rudder and I are chairing the session 3 presentations. The topic of session 3 is pest challenges to sweet potato production. The first presentation will be given by Dr. Rudder, and the title of that is Developing Tools to Manage Guava Root Knot Nematode, or Guava Root Knot Nematode in Sweet Potato. Go ahead, Will.
Thanks, Libby. As Libby said, my name is Will Rudder. I'm a research plant pathologist down in Charleston, South Carolina, the U.S. Vegetable Laboratory. And my primary job is to develop tools and knowledge to help vegetable growers manage problems with nematodes. And today I'm going to talk about a specific root knot nematode, the guava root knot nematode, Melodigine Entrolobiai. I'm going to talk about a little bit why it's such a problem in the U.S. sweet potato industry. I'm going to tell you about what we're doing to try to manage that, help growers manage that. Ooh. There we go. So sweet potato growers and vegetable growers in the U.S. been battling root knot nematodes, 3 or 4 species for over 100 years now. And more recently, we've had a new player come into the game, a new species come into the region, Melodigine Entrolobiai. It looks just like other root knot nematodes. So if you look at a microscopic image of the juvenile nematodes, even I, as a trained nematologist, can't tell the difference, can't tell you that this is Melodigine Entrolobiai. I need to do an actual molecular test to distinguish this species from those species that are already native and present in the Southeast. Unfortunately, though it looks the same, it acts very differently in the field. So the resistant sweet potato cultivars, such as Covington, that have traditionally been used to manage the endemic species, are infected and damaged by Entrolobiai. And so this nematode causes severe root galling, resulting in yield losses and severe storage root quality, lowering of storage root quality. So it has a wide host range, and it's hard to rotate out of this. And you can see at the bottom here is a infected fresh market sweet potato with the characteristic pimple and galls caused by this nematode. I'll talk a little bit. So as I said, it's a very new nematode relative to the native. It was only found in the U.S. In about 20 years ago in Florida. In North Carolina, it was only found less than a decade ago, actually on cotton and soybean. And since then, it's spread throughout most of the sweet potato producing counties in North Carolina and become a serious problem. This is likely because sweet potato is a propagated crop. So we think this relatively quick movement of this nematode to new areas is probably through the movement of infected propagation material, so seed roots, slips, as well as the movement of dirty farm equipment carries this nematode. So more recently than that, we were called up to a sweet potato producer up in Darlington County, South Carolina, where we thought we had a find. And although we had a nice big shed full of really nice storage roots that he was getting ready to sell, we started looking at in his call bin, and he was it was clear that he had a low-level infection of root knot nematode based on the galling on these potatoes. When you open these up, each one is filled with a bunch of female nematodes. And so when we took this back to the lab, we ran the molecular test and confirmed it was Enterolobiae on this purportedly root knot nematode resistant sweet potato. So the first thing we want to find out is, well, can we find any resistance to this nematode? So what we did, we looked to the USDA germplasm collection. We selected 93 PIs from that collection and ran repeated tests, inoculating Enterolobiae in the greenhouse, waiting 8 weeks, and then washing the roots, scoring the roots for the level of galling, and then actually extracting the eggs and counting each of these, how well this nematode is able to reproduce on each of these PIs. And the results were actually very good compared to other crops. So you can see this distribution of the different 93 PIs we tested. Covington and Beauregard here on the right were used as controls. You can see they average over 50% galling and high level of nematode reproduction, so they're very susceptible. But fortunately, we were also able to find 19 lines that showed a high level of resistance, so very little galling and almost no nematode reproduction. And this is great news for the industry. So moving forward, we're actually collaborating with the breeding programs and NCSU and LSU and trying to move this resistance into new varieties of sweet potatoes that have all the nice horticultural traits, the nice quality traits we've been talking about, and will really provide stakeholders with something to help start managing this nematode. And our own program will be screening new advanced clones in the coming months, and hopefully have something to move into trials later on this year. So resistance isn't the whole story. It's not going to fix the problem of Enterolobiae. Enterolobiae is unfortunately here to stay. We also need to provide stakeholders with more resources by slowing the spread and giving them a better management integrated pest management program to help manage this nematode. So one of the, as I mentioned before, it's likely that the movement of infected propagation material is responsible for the quick spread of this nematode. So on the left here is that infected fresh market sweet potato. I actually picked this up at my local Piggly Wiggly here. And like I said, I had to take it back to the lab and run the molecular test to confirm it was Enterolobiae. And then I also extracted the eggs from this and threw it onto a clean, some growing sweet potatoes in the greenhouse. And sure enough, it destroyed this. The nematodes from this potato destroyed this Covington sweet potato. So you can imagine if this was used as a seed potato and put into somebody's field, this is like a nematode grenade. It makes it into a field. That field is now contaminated for now and forever. So we really have to come out with a new way to slow the spread of infected material. And as I said, the traditional method of plucking out individual nematodes, I can confirm that individual nematodes are Enterolobiae, but I can't really tell you that there's not Enterolobiae, some worm, hiding somewhere on a specific potato. So to try to develop this type of a method, we turn to a commercial vegetable peeler. So we've actually developed this method where we can take uninfected sweet potatoes here on the left versus Enterolobiae infected. So we can throw one small infected potato in with a bunch of uninfected, actually peel them, collect the peelings that hold where the nematodes normally reside, and then run our molecular test on the skinnings themselves. And by doing this, we can say that a specific batch of sweet potato is probably contaminated or likely clean, free of Enterolobiae. So we found it to be a very sensitive method and we're hoping to continue valuing that in this summer. So to kind of summarize all this up, we're in the process of developing, working with other programs to develop M. enterolobiai resistant sweet potatoes that can be used by growers. We're also developing this new detection method to help. We're hoping that can help certify roots as clean or free from this nematode. We've also come together with other nematologists from Clemson University, University of Georgia, North Carolina State, to put together this project specifically focusing on enterolobiai. So it's Find ME. So if you'd like to know more, please visit our website at findmenematide.org. You can get updates on our research or information about enterolobiai in general. Our funding sources have been through the USDA-ARS National Plant Disease Recovery System, as well as NIFA Specialty Crop Research Initiative. And with that, I will pass it on to the next speaker.
Great, Will. Thank you for the great presentation. Had 40 seconds to spare. Piece of cake, huh? Hold on a minute, folks. I'm having some technical problems.
Dr. Williams, do you want me to bring it up on my side?
Yeah, if you could. Thanks, Sandy. Okay, great. Can you hear me?
Yes.
Yes, we can.
Great, thanks. Sorry about that. Well, I wanted to thank Mukareb and Phil for the opportunity to speak today about the work that we're doing. And what I'm going to focus on today is one of the insect pests that's really important for sweet potato, and that's a group called wireworms. And here you see an adult, what we call a click beetle. This is the adult form. This is the larva. This is the immature form that lives in the soil and is also called wireworms and causes the direct damage to the storage roots that cause this typical pockmarked damage that we see. Sandy, am I going to have to ask you to change slides? Okay, thank you. Here we see in this figure just a really quick diagram of the life cycle of the wireworm. Eggs are laid on the soil, larvae hatch, feed on the underground portions of the plant, pupate in the soil, and then adults emerge and live above ground, but they don't really cause much damage aside from the fact, of course, that they procreate and cause future generations. So wireworms are increasingly, the control is increasingly problematic due to the lack of effective management strategies. And part of this is because of the fact that they live in the soil and they're hard to control for that reason. They're also hard to study. Okay, Sandy. And at the same time, though, we know that behavior modifying chemicals have been used for a long time, more than 50 years, to monitor and manage insect pests of agriculture. And when I say behavior modifying chemicals, what I'm talking about are compounds that are produced by one sex of an insect and that affect the behavior of the other sex of that same insect species. However, we really don't know much about how this system works in the reproductive behavior of click beetles, okay, and how we might be able to use that to the benefits of agriculture. Okay. And so what we did was set out to do some work, and I was working on this, leading a group with Jackie Serrano. Jackie was a PhD student at UC Riverside at the time. She's currently a research entomologist with ARS. Paul Johnson, a professor at South Dakota State, and Jocelyn Miller, a chemist at UC Riverside, and he was Jackie's PhD advisor at the time. So our goal was to identify, synthesize, and field test female-produced chemicals that might be attractants for the corn wireworm. Okay. So here's what we did. To start out, it's pretty simple. We went out and collected insects. We did this by a variety of means: light traps, other traps, direct observation in the field. The objective was to collect live, adult, and larval corn wireworms. The next step, go ahead. The next step was to do volatile collection and analysis of these insects. Okay, what we did here was basically collecting the odors, the volatiles that are being produced by the insects, and then analyzing chemically to determine what those compounds are. We're not going to get really heavy into the chemistry at all, but that's what we were doing. The next step was to take those compounds that looked interesting that we learned about here and do electrophysiology. So electrophysiology is basically the marriage of chemistry and insect physiology. And it was a way to determine if an insect could perceive the compounds that were being produced and we identified up here. So, and you see here, I've circled this bit in red. This looks like an interesting compound that we wanted to follow up on. Okay. So we did that, and the next step was to do some syntheses. Okay, so we went in and did some more fancy chemistry in the lab and basically synthesized these compounds that we thought were putative sex attractants. And here's one we came up with. Okay. The next step was to take those compounds that we synthesized and go back to the field, all right, and set up traps like you see here in the field to see what we caught, to see what insects we collected. Okay, so here's what we discovered. We found that a single compound, a single chemical produced by female corn wireworm click beetles was strongly attractive to the male beetles. And you can see that here in this graph. This happens to be a, I think a dose response test, but you can see at all the concentrations, they were significantly more than the control. This is the structure for the compound. And here's one of our treatment vessels from, these are all male corn wireworm click beetles, about 90 of them from one trap from 1 week. Okay. Okay, so now what? Well, given that the sex attractant consists of a single compound and that it can be readily synthesized, it seems to have, there's a lot of potential to develop this for monitoring and management of corn wireworms. At the same time, we have to understand that there are other click beetle species that are also pests in sweet potatoes. And so at the same time we're working with corn wireworm compound, we're also looking for sex attractants for these other click beetle species that are also important in sweet potatoes. So that's sort of a two-pronged approach or an ongoing process. Next. But for now, we're going to focus on how to apply this corn wireworm sex attractant. And there are different ways to do this. I just want to touch on a few of them today. One is looking at trap design and placement. The idea here is to make a monitoring trap or to devise a monitoring system that is more user-friendly to crop consultants, scouts, producers than what we've been doing. These traps, this black trap here that we've been using is great for research, a lot of attributes there, but we think that maybe something more simple like just a yellow or white sticky trap that a lot of us are familiar with might be more suitable. So we're doing some work on that. I did some work on that last year in Charleston, and it looks pretty promising that the results that we see here are going to be transferable to a more simple, more usable, user-friendly type of trap. So that's promising. Next. And I should say, I won't go back, but I should say we're going to continue that work this year in the Carolinas and Virginia. So another approach we want to look at is to try to use the sex attractant to evaluate agricultural practices on cordonworm abundance and damage. And the project we're working on here is with Anders Huseth at North Carolina State and Tom Coor at Virginia Tech. And really Anders is leading this project, and I just try to basically stay out of the way. But the idea is to try to use the pheromone or the sex attractant that we have to evaluate environmental effects and cropping patterns to better understand corn wireworm damage in the Carolinas and Virginia. For example, environmental effects, soil type, other things, weed management practices, past cropping history. The fact that we have the sex attractant now is a valuable tool to evaluate some of these other factors to better guide our production of sweet potatoes in the Carolinas and the Southeast in general.
2 minutes, Dr. Williams.
Okay, while I was talking fast, I'll slow down. I have one slide left. Okay, Sandy. And one other possible approach to this is mating disruption. Okay, and then we see, and if you look in the upper left part of this figure, I don't know if my cursor is probably not working, but you see the typical sort of system where you have a moth. This is a moth, probably a cobbling moth. Yes, thank you, perfect. Let's say perched on a branch, she's calling. In other words, she's producing a pheromone, an odor into the field. It's a plume, just like as you would imagine a plume of smoke. The male senses it with his antennae, flies upwind, and they mate and procreate, and the population goes on. Well, what if, using this mating disruption approach, what if we flooded the environment with the sex attractant by putting out synthetic lures all in the field to basically disrupt the ability of the male to find the female? This is called false trail following in some lexicon, and thereby reduce the population level of the pest. This has been used pretty successful for some insects, cobbling moth and some others, and it has potential, I think, for click beetles as well. There are other approaches that I haven't mentioned. We can talk about that later in the Q&A session, but for now I'll stop, say thank you, and apologize for the technical difficulties that I had. Thank you. And with that, we will move on to the next presentation by Dr. Kai-Shu Ling. Dr. Ling is a research pathologist here at the Vegetable Lab in Charleston, and the title of his presentation is Importance of Management of Sweet Potato Viruses. Take it away, Kai.
Kai, are you there?
Sorry, I had to keep this voice. Okay. There you go.
Great, you're on.
All right, thank you. Thank you for the opportunity to share our work with you this afternoon, and appreciate the opportunity. And for this work today, what I'm trying to do is to share some of the highlights. And this is the work that is collaborations, long-term collaboration with my colleagues, Dr. Alvin Simmons. That we have been he's a research entomologist and on whiteflies, and myself as the biologist. And so, and also has the technician, the on-jack area, and along with many of the past postdocs, visitors, and technicians. So really appreciate their help. So this work here, we're looking at the sweet potato leaf curl virus disease complex, and looking at as to a complex system actually from the insect, the vector, and from the virus, insect, and the host interaction. So actually kind of looking at in the three way of the pathogens, the whitefly, the vectors, and then obviously the sweet potatoes. So what we kind of looking at is the sweet potato cultivar decline has been a serious concern to the sweet potato industry. Certainly there's some genetics, somatical some clonal variations, but the viruses are considered to be the major cause of the cultivar decline. And when I started in these projects in 2004, and there were several of the work has been pretty good work done on the aphid-borne putiviruses, like a sweet potato furry mottle virus. But there's this new emergent disease called in the whitefly transmitted bigomo virus. It's called sweet potato leaf curl virus. It has very little information. So we were kind of initiating this work and just kind of showing the kind of quickly on the sweet potato, the leaf curl, you can see that it's a leaf curling and then healthy leaf. So it's quite obvious when you're actually showing the symptoms. And it's whitefly transmitted, and this whitefly is global pests and really can affect in just sweet potato, but also have a number of the other crops as well. So our efforts was trying to actually first to, when we're taking this and working with my colleagues, the Bill Winamano, and we're leading the efforts to getting the international consortium of the scientists and sequence the first whitefly genomes called Ming1. Actually, this is used to be B biotypes, so you're probably more familiar about that. So we are kind of working with the Alvin Simmons. Actually, he has the ISO lines, and we'll be able to selecting these males only the population, which is just only the more the single genomes and copy of the haploids of the genome. So it was kind of easier for us to sequence rather than the female one's bigger size. So so that was kind of a breakthrough, and we are using this technology now, using this fundamental knowledge about the genome and developing RNA interference technology still going on in the lab. And so this is one of the looking at the sweet potato, the bigomo viruses. When we're talking about sweet potato leaf curl viruses, looking at it's just only one species, but it's several viruses, or we call the sweet potato bigomo virus is actually considered to be called sweetpo virus because it's kind of quite unique on the sweet potato when compared with other bigomo viruses on the genetics analysis, and it's still kind of quite widely distributed around the world. And obviously you can see here in the southeastern United States quite a problem on this sweet potato leaf curl virus disease. And here just kind of listing of some of the number of the viruses, and that kind of showing you why it's so important to study the genetic diversity of these viruses. So it's not just only one particular, what we call it, the quite unique uniform genetics, actually at least 11 and different viruses around the world, and at least in the US had 3 of those. Even some of those were considered to be called the sweet potato leaf curl South Carolina virus, because it's something from recombinants between the sweet potato leaf curl and then sweet potato leaf curl Georgia virus. So it, by understanding these genetics information, we'll be able to develop these conserved sequences for developing the real-time PCR testing, and which is the one that being used by the National Clean Plants Network as a standard method for detection. So that was, and I just want to point out that this is in collaboration with colleagues from the Alcorn State University. So it's important that you have that partnership with those people working in the Mississippi, and we collecting and then publishing this work with together with the colleagues from Alcorn State. And these the sweet potato, not just the virus is not affecting the sweet potato itself, but also affecting a lot of the morning glories. As you can see here, even when the sweet potato productions can pull off these weeds, the morning glories still can be the host of hosting those viruses, and whitefly can coming in, spoiling those viruses to the sweet potato when they are when you're getting the sweet potato field productions here. So that can be a problem. And so kind of turning to other aspects that we're kind of looking at just right now, many of these work being done by National Clean Plants Network. in most of the states, the major sweet potato producing states, they are doing great job of creating the clean plants network. And that's most of those are just for the virus infected plants. So it's kind of important to keep those in a higher clean plants and getting higher production. So in collaboration, our colleagues here in Charleston and the NC State clean plants actually developed generating from there. And we evaluated in the field trial and showing that several cultivar that we cleaned plant and then the virus infected one. And for example, these Beauregard's, 2/3 of the yield get lost by just single virus, by sweet potato leaf curl virus infection. Or the Sumer, again, 60%. And then the Liberty and Picadillo, about 1/3 of the yield actually lost, just had one season. So they're kind of showing that when you mentioned about breeders and spending so much effort to breed for the new cultivar, and you just have one season of the virus infection, the yield would be significantly dropped. That's
Dr. Ling, 2 minutes left, please.
okay. Cultivar decline, so that's why the name was coming from. So another project was a report coming up from the South Korea about the potential seed transmission of these viruses in sweet potato, and that was kind of a concern to many of the breeders and then the industry, because it's even though it's not from the true seeds, actually it's more focused on the breeders, but affecting the seeds imports and then exchange between the breeders in the different states. So we put a lot of efforts with our colleagues to looking at this and grow out and then showing that it's not really seed transmission. It's actually the white fly transmission. As you already see this picture from Dr. Wallows, kind of showing that the seeds, true seeds producing, and we actually working with the group on breeding and then looking at the seed grow out, as you can see here, in the greenhouse, in the grow chamber, and then different conditions, and then we show it's not the seed transmission, but it's really the insect transmitting of this disease. And with insects, white fly, it can actually cause these transmissions, leaf curl occur. Okay, so what we learned on the impact, improve these real-time PCR detection, which has been a standard method for the sweet potato leaf curl virus detection in the National Clean Plants Network. And the first genome sequence was obtained, and then which was actually extended into sequence the cassava whitefly in Africa. And when we had this support from USDA-ARS project, so it's really kind of important. It's a whitefly affecting cassava productions and transmitting several viruses, really devastating cassava production in Africa. And we also set this record straight about this no seed transmission of the sweet potato leaf curl. So that's just mentioned about it. So what we future plan to support the breeding efforts on sweet potato with resistant to sweet potato leaf curl virus and other viruses. So the breeding efforts, it's great to see the Dr. Wallows project, programs really growing, and we would like to kind of help contributing on that. And the sweet potato is the endemic here, so a number of those selection lines could have potential resistance already to this virus. So what we're going to next is screening and evaluating this cultivar germplasm for resistance, and also try to improve the phenotyping efficiency used through the machine learning and artificial intelligence. So that's all for me. Thanks.
Great, thank you, Kai. Right now, I'm going to hand over the facilitator duties to Dr. Rudder. Take it, Will.
All right, thanks, Livy. Our next speaker is Dr. Mac Utali. He's an assistant professor at Clemson Coastal Research Education Center, which is co-located with the U.S. Vegetable Laboratory in Charleston, South Carolina. And I'll let him take it from there.
Can you see my screen?
Yep,
Okay.
sound good.
So for the sake of brevity, this talk's really going to focus on emerging herbicide strategies in sweet potato. The plant architecture has already been highlighted. We also have some projects looking at allelopathy and some projects in collaboration with ag engineers and robotic companies trying to develop robotic weeders. But I want to talk a little bit about a compound, bicyclopyrone. Prior to coming to Clemson, I was with Syngenta Crop Protection in their herbicide development program. And this was actually the first molecule that came across my desk, or the protocol for evaluating the molecule came across my desk, and did probably about 100 or so trials with corn herbicide products. But it seems like Syngenta is interested in registering bicyclopyrone in sweet potato. It would be the first triketone herbicide registered in sweet potato. Then these types of herbicides inhibit an enzyme called HPBD, which is involved in carotenoid biosynthesis. And what's interesting about bicyclopyrone is that it is more active on grasses due to this 2-carbon bridge structure here, relative when you compare it to some other triketone herbicides. And then this hydrocarbon tail right here has been optimized in terms of its number of carbon length to provide about 6 weeks residual control. Now, certainly going to be dependent on soil type and soil temperature, but that's a really good control window for residual herbicide if you're potentially worried about carryover concerns. It's translocated through both the xylem and phloem, so it would be there's some application scenarios where you could apply it. Weeds could emerge, and then those weeds could still be controlled by taking the herbicide through the roots. And I've done first time I saw it was in sweet potato, was the trial in New Jersey, and it looked pretty good with their weed pressure. They have lamb, I think it was some curly dock, goosegrass, lamb's quarter. Look at the untreated, you compare that to bicyclopyrone applied pre-emergently. However, the driver weeds down in the south, yellow nutsedge, palmer amaranth, bicyclopyrone is not going to be a standalone herbicide. So it'd have to be in combination with something like a reflex, a famecifin, a dual magnum. But it would be, adding those 3 compounds together would provide a really robust pre-emergent weed management program. There's not really many options or any options for selectively controlling yellow nutsedge in sweet potato post-emergently with the herbicide. So we did some herbicide safener studies, looked at some compounds, some brassinosteroids as well as melatonin, and tried to safen this herbicide Bazagran, where the active ingredient is bentazone, which has activity on emerged yellow nutsedge plants, but doesn't have much soil residual activity, so carryover wouldn't be too much of a concern. We did an in vitro and greenhouse study, and there were some promising leads. You can see this picture here is bentazone applied alone. You look at bentazone with melatonin, and there's less of that kind of chlorosis, which are indicative of injury from photosystem 2 inhibiting herbicides, which is the mode of action of bentazone.
2 minutes.
2 minutes, yeah. So we took this study out to this kind of concept out to the field. We had looked at bentazone applied alone, melatonin alone, Vitazyme alone, which is a product which contains a couple brassinosteroids, and then with Bazagran or bentazone. And we did one study at a commercial field. You have on the y-axis injury in sweet potato attributed to bentazone. The dark green is bentazone alone, and then these colored bars are the bentazone with some of the plant hormones. And you can see 3 weeks after treatment, there's a significant decrease in injury in bentazone, and then 6 weeks after treatment, using the plant hormones reduced injury. And there's an interesting phenomenon as well with weed control. So bentazone is going to provide probably max 85% weed control. So if we noticed an increase in weed control with the plant hormones, that wasn't because the plant hormones were making the bentazone more herbicidal. It was that the bentazone and sweet potato alone plots, there was injury, which reduced the canopy closure. So the yellow nutsedge that didn't get killed off was able to recover and grow through, whereas the plots which receive the plant hormones, the canopy was able to close and help suppress yellow nutsedge. And then looking at yield in the commercial field, you look at the non-treated check. So it's not a true non-treated check. In this commercial field, they used Reflex Pre, which is a really robust pre-emergent herbicide. It's a good, these weren't that weedy plots. There's just some nutsedge popping through. And then you look at the total marked yield was almost double when you increased, when you added the plant hormones to the bentazone tank mix. And then, when you compare it to reflex alone or reflex plus bentazone without any of the safeners.
Okay, we better finish up.
So yeah, so it's similar concept in Charleston Fields. You can see the pictures here. Bentazone chlorosis with the plant hormones, canopy closure, and then this is at Charleston. You see bentazone yellowing of the leaf tissue with the plant hormones. Clean plot, but healthy sweet potatoes. So that's it. I know if Katie Jennings is on, I know she's done a lot of work with bicyclopyron and sweet potato in North Carolina. And then next steps would be to contact the registrant for bentazone, which is Winfield, and prepare a data set for them. We are repeating these trials in South Carolina this year as well. So thanks.
Great. Thank you, Dr. Telly. If you could stop your share.
Sorry.
And Dr. Quesada.
You ready for me then?
Our next speaker is Dr. Lina Quesada Ocampo, Associate Professor from North Carolina State University.
Thank you so much, Will, for the introduction, and to the organizers for the invitation. Can you see my slides okay and hear me okay?
Yes, we can.
Excellent. Well, I only have 5 minutes, so I'm going to keep this very short and sweet. So I'm the vegetable pathologist at NC State, and one of the crops that I cover, lucky for me, is sweet potato. So these are some of the diseases that we worry about in North Carolina that I get calls, every year about. So Melodigine Entrolobiai, of course, which Will already talked about it, scurf, black rot, southern blight, fusarium root rot, rhizophus soft rot, Deutericum sour rot. Some of these are seed problems. Some are field problems that progress post-harvest, and some are exclusively post-harvest. Something that really caught my attention when I started working from sweet potato and coming from our vegetable crops is, one, outside of viral pathogens, we don't know a lot about sweet potato pathology, like very foundational biology knowledge of a lot of sweet potato pathogens is just not known. Second thing is we don't have a very robust chemical control toolkit, not like other vegetable growers have. And those two things, I think, make us a little vulnerable in the face of a disease emergency. And I think that became painfully obvious when a few years ago, we had some issues with black rot. I was very new to my job, and we had a very significant black rot outbreak in the state, and it spread to other states, of course. We were able to stop it fairly quickly thanks to a Section 18 emergency label with an effective fungicide that we identified. But I think that that emergency really highlighted that we needed to do more. The industry, because we have a fantastic industry in North Carolina, came together and actually funded a 5-year project in my lab to do exactly that. So for 5 years, we have been studying black rot, a lot of it in collaboration with other labs. Most of this work was done by my PhD student, Madison Stair here. And she has been asking questions regarding dispersal, survival, how to manage it, crop rotation, host resistance, all of it. We have made a ton of progress there, which is great. Also, something really good that came out of it is NC State, I think, really also saw the need to invest in this area. So we secured some permanent pathology resources from the university. They built us a post-harvest facility in one of the research stations, well away from the other programs so that we didn't get pathogens in like the breeding lines or things like that. But it's really nice because we have a mini packing line, we have curing rooms, we have disease nurseries, and that really has allowed us to make just really fast progress, which is great. So thanks to those resources, we have leveraged, obtaining federal funding to work on this, which we have been really lucky with that. So recently we have secured now 2 grants from the Foreign Ag Service to work on sweet potato trade barriers and exports. So something that has been happening in recent years is that, we have very limited chemical control options. And then those options have become threatened because of changes that have arbitrarily happened in just pesticide import tolerances in our export markets. So these projects are really trying to find alternative management strategies from cultural to chemical. And several folks from NC State are involved. In entomology, we have Anders Huseth and Matthew Bertone. Nematology, Adrian Gorney. With science, Katie Jennings. And from horticulture, Jonathan Schulteis. And it's a really nice partnership with IR4, which recently moved their headquarters to NC State. So what we're trying to do with this next 3 years that we have left of the project is secure as many pesticide registrations in sweet potato as we can to be able to address just disease issues.
2 minutes.
Thank you. So I'm really hoping that at the end of the project, we would have made some really great progress there. So that said, I'm a big believer in looking at the long term. Most of what we do in my lab are really short-term things because we need to keep our farmers farming, but breeding is really important. And a lot of the ultimate solutions to this disease issues are going to come from breeding. But breeding is really hard and really slow, especially in an exploit. So something that we have been trying to do on the pathology side to support our breeders is trying to generate a catalog of resistance genes in sweet potato. Essentially, we're sequencing the sweet potato resistome and several sweet potato lines that breeding programs are giving us. And the idea is that when that catalog is ready, we can not only use traditional breeding, but also functional genomic approaches to quickly identify our genes that are key. So that then our breeder partners can more quickly introduce those genes to preferred lines, which would be great. Host resistance will also have the advantage, of course, that it can circumvent the issues with import tolerances. And the catalog can be applied to any disease that there's a effector-mediated response, because these are our genes, NLRs. And then last, I'm not going to talk a lot about this because we already did, but we already are also part of the Find Me Melodigine Entrolobiai SCRI. And in NC State, the team is myself, Dr. Adrian Gorney, and Lindsay Thiessen. We're focused on sweet potato and rotational crops, mostly providing support in diagnostics and distribution on Adrian's side, and for me, just management. And that's what we have been up to. And with that, I'll be happy to take any questions or comments later in the discussion section. And thank you so much for your time.
Great. Thank you, Dr. Quesada. I think next is our break session, correct? Our listening question or comment section?
Right. Thank you so much, folks. And thank you, facilitators, Dr. Rudder and Dr. Williams, for a great job. And thanks to all the speakers. Let's give them a big virtual hand, folks. Are there any questions from this panel at this point? We have about 3 minutes to our next session. I guess there are several questions online. For example, if you don't mind, I can read one. In Africa, cost of pheromones, attract and kill, have been too high to facilitate use adoption. Is this an issue in the United States?
Yeah, it's a good question. It's a good question. I think it depends on a couple of things. Number one, what crop is it and the value of the crop. It also depends on the chemical itself. Some things are just easier to synthesize. We got a little bit lucky with the corn wireworm compound because it's just one compound. It's not a blend of several. And that compound, so that simplified things. And that compound is also, relatively easy to synthesize. But again, I think the value of the crop plays a big role in that. And I think for a crop like sweet potato, that in Africa and Southeast Asia is very much a subsistence crop. The pheromone technology can be a bit of a hurdle to clear. And so, yeah, that's an obvious hurdle.
Okay, thank you. Another question. Are there any plans to use irradiation to prolong the shelf life of sweet potatoes, especially for export?
May I ask which kind of irradiation we're talking about?
Well, the question did not say, but says using irradiation.
Oh, well, there you go.
How
Thank
about
you for-
UV? Says
So
UV beta.
I actually have done a little bit of research on it, and it does help a little bit. We did not continue that research because we couldn't figure out a good way to like implement in the packing line, but there is some effect, for example, to protect the sweet potato skin from rhizoposol rot. We have documented that. That's what I was curious which radiation people were asking about. Thank you.
Okay, great, thank you. I guess we can refer most of the questions to the next session because they're more related to comments than questions. So at this point, let's again thank the panel and get ready for the next one. The next panel, which is coming up at 3:30, which is right on the dot, is to listen. Folks, you know, when I was brainstorming with my team, what kind of sessions or programmings do we have? The very first thing which came to my mind was that we not only would like to share what we have going on in ARS and our collaborators' world, but also it's very important for us to listen to what our stakeholders expect of us. Us means researchers in general. I would repeat something which my boss just told me a few weeks ago, that Mukarrab, if we are not relevant to our stakeholders, we have a problem. So here's an opportunity for us to listen to our stakeholders, and who best could be to lead that conversation other than Michelle Granger, who is the Executive Director of North Carolina Sweet Potato Commission. She recently organized a beautiful stakeholder, growers, producers, farmers, industry folks session, which I really enjoyed very much. She's a pro in doing this. So Michelle, if you are on.
I am here, Dr. Qureshi.
All is yours.
And my gosh, you have set the bar high. So
very high for a purpose.
Thank you so much for the warm introduction. And thank you to the entire committee for this incredible event. And most importantly, to all of the presenters and each of you who have very much been engaged this duration of this program, and the level of interaction and questions you've posed have been fantastic. As Dr. Qureshi said, it is my responsibility for this last session to help facilitate in the listening. So this is really more about, for those of you who are still with us, to pose your thoughts, where your needs you have identified are for the industry, and how the different stakeholders that I think have been very much equally represented in participation as well as in presentation can help make those needs addressed, help meet them for you, really. And if there's gaps, how do we fill them? So let me just say, I have several questions that I can address because many of you were so kind to fill out the pre-symposium survey. However, I'd like to give you a chance to pose some immediate questions that you may have come up with while we were sitting here and throughout this symposium that you would like to ask. And I'm going to lean on Dr. Qureshi and Sandy to help me monitor the chat feature and hand raising while we go through this.
Absolutely, we will.
Great.
We'll be delighted to.
Wonderful. So while I give you a chance to type some questions, because I know that does take a moment or two, I'll remind you of the 5 questions that we asked in the survey. First, what additional resources, information, research, marketing, etc., do you feel the sweet potato industry is currently missing? Second question, where are your biggest challenges in the sweet potato industry? What would you need to expand your sweet potato market? How important is consumer preference, culinary quality and variety success. And finally, and most importantly for this symposium, how can we best provide future engagements between sweet potato stakeholders and USDA-ARS? The survey netted back quite a few responses. And if I circle back to the very first question of what additional resources and information are you looking for, there were quite a few that I can answer from the North Carolina Sweet Potato Commission's position. Let me start with one inquiring as to, have we ever looked into featuring sweet potatoes, specifically North Carolina sweet potatoes, in television? The question focused on local, but the truth is we have several projects that are ongoing right now that are going to net themselves for a much broader audience. So from the local perspective, real time, Spectrum News here in the Triangle is doing a series of features on the North Carolina Sweet Potato Commission and our growers in the industry as a whole. The first aired a few weeks ago, and the second will be airing mid-May. We are posting those on our YouTube channel, so and linking them, excuse me, we're not able to post them on the YouTube channel. We tried that. We're linking them in our social media accounts. But the whole idea is to recognize the Commission's 60th anniversary and the great history that our growers have provided for this industry. Additionally, we have a opportunity to participate in a new national broadcast that is going to be produced by Chip Carter, called Where the Food Comes From. RFD TV is going to air this broadcast of an entire series of all about where our food comes from. North Carolina sweet potatoes will be featured in season 1. And once RFD TV has aired it, that will then hopefully live permanently in streaming services, whether that be Amazon Prime, Netflix, or other type offerings. And we are also creating real time right now, a year in the life of a sweet potato. That's a project that will indeed live on our YouTube channel, as well as our other social media, and is all about the educational aspects of our great industry. So we're super excited to say that, yes, currently we are trying our best to get the good word out about our state's superfood. Another question that came in was, how are we reaching our consumers and educating them on the versatility of the sweet potato? Well, I appreciate that, because this office, we are doing quite a bit in that effort and endeavor. You saw several presentations talking about French fries and potato chips. So through the benefit of tying together our researchers and what they are learning in their variety trials, and then taking our efforts in marketing and promotion, we are able to work with all kinds of influencers, chefs, restaurants, and other dietitians. That should be noted that we do have a dietitian of record, registered dietitian of record for the Commission, and we work with others throughout to truly show the well-roundedness of the sweet potato and just how versatile it is, and in how many applications it can be used throughout the entire day, starting with breakfast and finishing off with a decadent dessert in the evening. And we've been working with those who are sports enthusiasts and specialize in nutrition and dietitians as well, to really emphasize the regenerative powers of the sweet potato pre and post workout. So I appreciated that those came through under that question. I know that it looks like Dr. Lester from the USDA-ARS is still on with us, and if he would allow me to impose on him, I would welcome the opportunity to ask him what his take is currently from a federal government's perspective for strengthening our overall portfolio in sweet potato research and efforts.
Well, thank you for that opportunity. Of course, we would like to continue to advance the food science and market quality aspect that has been woefully underfunded for a long period of time. But as Dr. Ellen Harris has mentioned, that's where we need our stakeholders to advocate on our behalf to Congress to increase our budgets there. And quite frankly, I would like to see a typical increase, and that would be about $1.5 million. So also in the area of quarantine and production and protection, we would always welcome an increase in that aspect as well. As you saw, there were a number of issues with the whole breeding, but great advances and opportunities to solve a lot of problems there. And then some of the more recent aspects with regards to nematodes. So there's many, many problems on issues that still need to be addressed from a research standpoint. And we can only do that as our budgets allow us to hire and expand on our research capabilities. But we want to do that, but it's a domino effect.
Got it. Thank you, Dr. Lester. And I hope that each of you on this call heard loud and clear that he needs us as stakeholders to help further advocate for additional funding. And I can tell you from the North Carolina Sweet Potato Commission, are in full support of such additional funding endeavors and are willing and ready to take what we need to do from our lead perspective to help do such that. And I look forward to linking arms with many of you to also raise that awareness and make that ask.
And Michelle, it's pretty simple. I mean, basically we have, all sorts of commodity groups have gone to Congress over the last years, whether it's sorghum or cotton or catfish or pecans. They work with their federal congressmen and primarily their senators. And they basically say ARS, and the language is simple. ARS, for an example, $1.5 million increase in post-harvest sweet potato research. That's as complicated as it gets. That tells us three things. One, ARS gets an increase of $1.5 million. Two, it's post-harvest, or it could be pre-harvest, depending on the language. And then three, because it's post-harvest and it's sweet potatoes, it goes only one place, and that's Raleigh, North Carolina. And then I, as the national program leader, I write the directive memos. That then goes to Ellen Harris in our Southeast area. They then make sure that they write the position descriptions to hire the scientist to fill that. And then I work with those scientists to develop their research objectives and their approaches so that we accurately reflect and address your issues and concerns. It's not a complicated thing at all. We just need the resources.
Fantastic. We will do our best, and thank you for spelling it out so clearly. I have see a couple questions and comments that are happening in the chat box. And I see one from Tawanda that reads, are there plans to work with school districts to have sweet potato on the school feeding and lunches? Well, Tawanda, I look forward to meeting you one day and giving you a high five for posting this in the chat, because just today there was a press release that was announced out of our office and is already starting to be picked up, that we have an entire food services video program that has been dedicated to our school food service professionals. It's a series of 4 videos that break up the purchase, storage, preparation, and general history about the sweet potato into different segments. Oh, I see Coco Daughtry, our communications specialist, has put the link in the chat for you as well. We're very excited about these videos, and they are a complement to a project that started before those videos, and that was the creation of different school dietitian recipes on the scale of school meals utilizing sweet potatoes. And these recipes range from quick snack-like foods to side dishes on the children's lunch plates, all of which have been hugely supported from two different groups, the North Carolina Tobacco Trust Fund Commission and the North Carolina Department of Ag's Farm to School program. I encourage you to definitely check those out and to share. Please share with others these resources, because we are very proud of them, and we are super excited to have the opportunity to introduce new generations to our North Carolina sweet potato and to hopefully hook them early on into their flavor profiles and all the different ways that they can be prepared. Thank you.
Michelle, this is Mukarab. One of the comments we got under question 1 is very, very interesting and very near and dear to my heart. It says, more great sweet potato industry trade association meetings with USDA lab in Raleigh. Isn't that what we are doing right now? I think this is fantastic that even our stakeholders are recognizing the value which this collaboration can bring. Researchers working hands in hands with commodity groups and stakeholders such as you. I would offer that if this helps the cause, that we make this as a yearly event, that we get together as maybe a small group, leave it to North Carolina. Maybe we can expand it nationwide, but I know you and Kay and other national sweet potato organizations do a fantastic job in bridging this gap and further supporting this partnership. But at North Carolina level, and remember, ARS is national. It's not just local, Raleigh, North Carolina, or Charleston, South Carolina. We have a national, even global footprint. So any ideas which, Michelle, you and your colleagues have, how we can better streamline this on a more regular basis, we are all game.
Fantastic. And I would say that we are also receiving feedback at the Commission's office of great interest in more events balanced between virtual and in-person, and special targeted topic events. So I think this falls perfectly under that. I've been scanning the participation throughout the symposium, and I'm so pleased that so many of our growers have made time to sit in and participate today. We have a great diversity of representation, as you've said, but today is a beautiful day here in North Carolina, and many of them have very diligently chosen to miss out on being outside and with their working on pre-season to be part of this. And I thank them for that. And I thank everyone who has been asking the questions and engaging as they have been. Are there any other questions before I tackle the additional survey responses that we've received?
Michelle, Craig here, real quick. I want to echo what you said before and just confirm with Dr. Lester and the rest of the ARS team. I think this is, Dr. Qureshi, this is a great idea, and I'm a really strong proponent of this. I can't tell you how important the food science aspect is now to the variety development pipeline. It's such a critical aspect of everything we do. So from a breeding perspective here at NC State, it's really a must-have. And I'm really working hard with the team to make sure that we continue that excellence that we had that was started that then kind of went over the past. I'm worried we won't be able to keep that expertise moving ahead. And it would really impact our programs and our ability, to understand the important traits, nutritional traits in sweet potato. And the other thing that I think that Mukarab said that really strikes a note with me is that they help us reach out to all of the ARS labs that have various competencies in the food science realm that we don't currently have. I'll give you an example. Purple flesh sweet potatoes, quantification of anthocyanins is not being done in-house right now in Raleigh. It's being done with another ARS lab collaborator down in Mississippi. And so they broaden our reach, and it's a really, really critical resource. So to the extent, Dr. Lester, that you can help solidify and increase this team here, I think it would really greatly benefit the whole industry, not only our industry here in North Carolina, which is of course really critical, but it will benefit the whole U.S. sweet potato industry. And I think it's a really important part to keep that growth going in the sweet potato side, the research side. So just I wanted to reaffirm that commitment from our side as to how critical you are to our partnership.
Well, Craig, thank you very much. And I'm certainly on board with you. I'll do whatever I possibly can, along with Joe Munyaneza and Tim Widmer, who are our two other national program leaders on our Zoominar today.
I know Joe well from my prior days in potato. Joe, I'll do anything I can to help you, man. Good to see you.
Thanks, Craig. Yeah, so I think everything has been said, and I echo Gene, what he mentioned. But again, ARS cross states. So, you have seen how much work being done, for example, in Charleston, which is really supporting the whole sweet potato industry. And so without that production, research to really increase production and protection of sweet potato, I don't think we're going to have anything to process, right? So it's a whole holistic, we have to take this holistic approach.
Absolutely.
Production, processing, and everything. So as far as the ARS is concerned, we cross the borders. So it's not really like when you're talking about universities, which are more kind of restricted to states, but ARS goes everywhere. And so we're here to help. Thank you for your support.
Michelle, I think regarding the request for additional resources, funding for our sweet potato research program at the USDA Food Science Unit. In the past year, almost every year, we work with the industry, and every year, I think there was also the group of sweet potato growers and Sweet Potato Commission and also U.S. Sweet Potato Council went to the Hill and just do kind of the, I don't know if we could use the word lobbying, other things. So every time, every year, I also presented to these U.S. Sweet Potato Council and also the NC Sweet Potato Commission for the need of more resources for our research. We requested for additional scientists to conduct more research. So every year, I think we started since 2005, 2006, and then after the visit to the headquarters in the Hill, I think the team also went to visit the ARS headquarters. I think meet with Dr. Gene Lester and others over there too. So, and then we also kind of communicate with the headquarters about that for support. So every year, and then after that, I follow up, after the, I think it happened sometime, usually in June, so I call and follow up, or even sometimes, the Sweet Potato Commission call me and say, wow, it's well received, well support, but it's not really time for additional funding because budget is always tight, so that's what we received almost every year in the past 10 or 12 years. So hopefully with the coming years, the request would be materialized, and then we're looking forward to see more additional funding, especially more, scientists to conduct the research on the food science, as Greg and others also mentioned. So looking forward to see more, resources coming to the USDA-ARS lab.
Absolutely. And thank you so much for that feedback. I can assure you that as soon as we are able to make Congressional Hill visits and things have opened up, I will be there with a delegation. They keep assuring me that we're doing that as soon as we possibly can. In the meanwhile, I definitely am able to have conversations with our representatives in their offices. And I absolutely will be carrying forward what we've heard today. And more than likely, we'll be making a few phone calls to some of you to help me deliver those requests by phone, at least for now, until I can get to DC myself.
So Michelle, I just want to say your effort doesn't have to go every year. I mean, all that we'd like it, but once you get a congressional appropriated funding increase, say of $1.5 million to the Food Quality Lab, that's it. It comes to us every year unless you want to go back and increase it.
Okay. Very good to know.
I like that increase part, Dr. Lester. For that, I wouldn't mind Michelle go back again.
But, the thing of it is, the current administration, and I don't know if there's any congressional people on this Zoominar today, but we're learning in the current administration that spurring rural economies is critically important. And by advancing more and greater production of high quality sweet potatoes does just that one thing. Through the research in the Market Quality Laboratory, it spurs rural economies. It's all connected, as Greg said.
Absolutely.
And I just have a message in the chat here from Kay Rensel, who's the,
oh yeah, Kay.
She's saying USDA-ARS team, the U.S. Sweet Potato Council continues to advocate for support via virtual means with all members of Congress. Thank you for what you do to support the industry. So this is beautiful. I mean, this is exactly in the line of what we have been talking about, that, yes, money is important, resources is important, but it's really the relationship and dependency, the mutual, the reciprocity. If we are strong, the industry is strong. If industry is strong, we are strong. So that's the spirit I'm hearing, and I'm grateful.
Well, Dr. Qureshi, I feel that I may have be bumping up on my time, but I definitely hear a spirit of collaboration as well. And I am very happy to hear that, as I'm a big believer in that is how things get done. And I feel that we have an incredible community in the sweet potato industry across borders, North Carolina and beyond, for sure. And I'm very, very happy to be part of it, and certainly to have been part of this conversation today.
Well, folks, thank you so much, Michelle. It's just about 4 o'clock, and that was our drop dead hour. Any last minute comments, folks? And I know there are several legislative staffers and agriculture liaisons. I would love to invite them to make any comment if anybody would want to make any comment at this point. I've been getting a lot of chat messages from those. So thank you so much, folks. If I could conclude then, first of all, my bundle of thanks to all of you, especially our stakeholders and collaborators who make it possible for ARS to address our national need, our national agenda, which is essentially not only to produce enough food for, in your lifetime and my lifetime, to be 10 billion people on this planet Earth, but produce nutritious food and reduce waste. I think in that sort of an alignment with our national program offices and support from our Southeast area leadership, we can't go wrong. So thank you so much, folks. We're, as I said, recording this meeting. We have probably hundreds of still unanswered questions in the chat box, which I will be gathering up. I would be shooting some of you with these questions for answers so that when we publish the transcripts of this meeting, we can not only include those on the website, but also my goal really honestly is that we follow up with you all those who have registered, we have your emails. We will send all these answers to your questions because the time is so short, we couldn't really take every question today. Joe, do you have a comment last minute? I see you on the screen.
I just want to say great job,
Oh, thank you.
This is great. Thank you.
Thank you. I appreciate you joining. And I know Charleston, South Carolina location speaks highly of your leadership. So Gene, you have your hand up?
No, I'm just saying the thumbs up. Great job.
Okay. All right.
Everyone.
All right, folks. You have a wonderful Dr. Harris. Thank you if you're still on. We really appreciate you taking the time out of your very busy schedule today and continuing to stay with us. So thank you so much. And I probably messed up on thanking Dr. Linton, the Dean of College of Ag and Life Sciences of NC State, and Dr. Steve Lammel, the Director of Experiment Station. I know they were on, I had seen their names, but I forgot to acknowledge you. So before we all log off, thank you so much, NC State, for being a great host to ARS Labs here in Raleigh, North Carolina. And Charleston location, thank you for partnering with us. It was a great endeavor. Let's keep doing it. All right, folks, have a great rest of the evening. Goodbye.